Maybe. I hope you both are here and doing well. The topic of my talk is artificial intelligence in clinical trials. Well, some people consider this as intelligent clinical trials. Is it really intelligent or not? I will try to explain you in the coming slides. So in the first few slides, I will try to explain the process of clinical trials so that you all can appreciate how AI is influencing the clinical trials. So this template is a very popular template. This shows the evolution of a drug. So many of us wonder why some of the drugs are very super expensive. The reason is what you see in the slide. This is a typical example, like during drug discovery stage, say if we have about 5,000, 10,000 compounds, which we predict have good effect for a certain disease or a condition. And then we take all this 5,000 to 10,000 drugs to preclinical stage. What we end up doing is like a lot of in vitro assays and in vivo assays. At the end of the preclinical experiment, what we would left out with is like 250 compounds. Okay. So not all 250 compounds can be tested in the clinical trials. So we pick the best 5 compounds that show the effect, and then we test them in clinical trials. And we submit the result of all 5 compounds. And then the regulatory authority will approve just one drug out of those 5 compounds. And if you look at the time span, like here at the bottom, you can see that a considerable amount of time is invested right from the drug discovery stage to regulatory approval stage, right? And then a lot of money are involved in it. So that's why you see some of the drugs are very super expensive. You should also understand that the methodology and the process of the clinical trial will differ based on the type of drugs getting tested. For example, drugs that are used for the treatment right, of a disease or a condition. So to do this kind of clinical trial, you need volunteers who are affected by the disease or that condition. Right. The second type of clinical trial is drug for prevention of a disease or an illness. Right, vaccines comes under this type of clinical trial. No wonder, like COVID vaccine is very popular and is in the news pretty much every day. For doing or performing this kind of clinical trials, what we need is healthy volunteers. So the third type of clinical trial is for testing the diagnostic kits. Right. So diagnostic equipments will be used to test the patient samples. So for this type of clinical trials, we need both healthy volunteers as well as non-healthy volunteers. Last but not the least, this type of clinical trial, the quality of testing the quality of life and supportive therapy is very popular in the US. These are for people who are experiencing anxiety, depression, relationship issues, and personality disorders. Right. So now I will explain what are the different phases of clinical trials. I'm sure many of you are aware that clinical trial happen in multiple phases. So the first phase is called as a phase 1, which is also called as early phase. The major emphasis in this phase will be for the optimal dose finding and safety. Right. So this phase will start with 20 to 100 volunteers, healthy volunteers. As I said, the major emphasis will be on dose finding. So safety is the major concern in this phase. Once the drug passes the phase 1, it enters phase 2. Phase 2 will have a significantly, Phase 2 will have a significantly increased number of volunteers or patients. A major emphasis of phase 2 will be on safety and a little bit on efficacy. What happens in phase 3 is that a large number of participants will be involved. The drugs will be tested on large number of population for a particular disease or a condition, and the major emphasis will be on the efficacy. Not to forget the safety too. The safety will be of equal importance to efficacy, but the major emphasis will be on efficacy. The data will be collected, the data will be analyzed, and then it will be submitted to the regulatory authorities, whoever it is. In US, it is FDA. In the European Union is EMA. So once this regulatory submission happens, the regulatory authorities will go over the data. They will check the analysis of the data. They will make sure the result makes sense and there's no bias in the result. And then once everything is done, and if they are satisfied with the result, they'll approve the drug for marketing, right? So once the marketing starts, the patients or the subjects who takes the drug will be asked certain questions by the company who makes the drug of any safety concern. And those data will be collected, and it's called as phase 4, which is post-marketing approval and surveillance. So as I mentioned, there will be plenty of data that will flow in from all 3 phases, even from phase 4, and it has to be organized or structured in a way that it has to be presented to the regulatory authorities. Right, a lot of data will come into the safety domain, so it has to be split in such a way that it is organized, and even the people who are analyzing it know which data is stored where. These are some examples of the data sets that we store the data or we organize the data. So all the lab results. Lab result is where, like, as the typical lab visit, even we experience this once in a while. We go to lab, we'll give our blood, and then serum will be taken, and series of tests will be done. For example, AST, ALT, other things like which can be taken from serum, urine, and all those stuff. What type of result is this? Whether it is AST, ALT, what is the value of it? Everything will be stored in this lab data. The second thing is vital signs. So vital signs is nothing but your height, weight, your calculated BMI, which is basal metabolic index, and then waist circumference. All those data will be stored in vital signs. So one of the most important data sets or the domains that the under safety is like adverse events, which is nothing but the collection of the side effects after the start of the treatment by the subject or by the patient, right? What are concomitant medications? Concomitant medications are any medications apart from the test or the placebo treatment that the subject is taking. A classical example is a subject may be involved in a diabetic study where he's taking either a placebo or a treatment for diabetes. But one day he's having a pain or a headache, he's taking a paracetamol or an ibuprofen. So those kinds of medication will come under concomitant medications. And the other important domain or data set is the exposure to study drug. This data will have like what kind of medication, the treatment medication or the placebo medication, or what is the dosage of that medication, what form is the dosage taken, whether it's a tablet or a liquid. What is the frequency of medication, whether it is like twice a day, once a day, or like whether it's like twice a week. So those kind of details can be found in this exposure to study drug domain. Similarly, in efficacy, giving example of an oncology study so that most of us can relate to it. Tumor identification, say the MRI result or a CT scan result will tell like what kind of tumor it is, whether it's a primary tumor or a secondary tumor, who's the investigator, those kind of data will be stored in tumor identification. Similarly, in tumor results, an investigator will analyze the tumor identification data and how long or what is the diameter of the tumor. And like change from the previous visit, whether the tumor has grown, whether the tumor has regressed, all those data will be found in the tumor results. The third is a disease response. Disease response is how the tumor responds to the treatment, whether it could be whether radiation or chemotherapy. There are different kinds of response in tumor. One is partial response, where the tumor is reduced to a particular size but not completely. Complete response where the tumor has regressed 100%. Stable disease where there is no effect in the tumor from the last measurement to the current measurement. progressive disease. The tumor has grown considerably bigger than the previous visit. So these are some of the examples of disease response. One of the important endpoints in oncology study is either progression-free survival or overall survival. So in case of overall survival, it's right from the start of treatment to the death. They will calculate the time. In case of progression-free survival, it's right from the start of the treatment to the first progression or death, whichever comes earlier. So the event will stop there, or the time will stop there. So they'll calculate the duration from the start of the treatment up to death, and then plot a survival curve to determine the efficacy of the drug. All right, let's go to the next slide. So I think I have explained like the clinical trial process like in the previous slides well, but to put that in a nutshell, this is what it looks like. So the clinical trial will start with planning all the necessary steps initially, like creating a protocol in accordance to the standard operating procedure of a sponsor or a company and getting it approved, right? Then they'll find a suitable investigator who has expertise in the field of the study. Then once the investigator is decided, the other process that influences the trial, like the country selection, the site selection, the vendor selection, and the subject requirements will all be finalized. So after this, the patient recruitment will start. So believe me, this is a very slow process. It takes a lot of time to recruit the correct patient, and most of the patients that comes in will have some other condition which will make them ineligible to participate in the study.
And these
so it's a very slow process. Most of the time in conducting the clinical trial will be spent at this step, the patient recruitment and participation step.
The rationale
right?
for
Once the patient recruitment happens, what happens is data will start to flow in. Most of the steps are automated now. So what happens is once the patient goes or a subject goes and visits a site, say for the lab, and once the lab releases the result will automatically go into the sponsors of the company's website. So once the data comes in, data will be reviewed for accuracy or any mistakes. If there are any mistakes, then all those data will be rectified. Data will go through the statistical analysis department. So this is where I work. I work as a statistical analyst. It's about collecting the data, organizing the data, and then to produce the necessary tables, graphs, and then submit it to the FDA or other regulatory agencies. So once we do the statistical analysis, we produce a table, we present it to the medical writing team, where they will write a manuscript, they will present the data in conference before submitting it to external agency for approval.
And the cell line modules
so we have after the step, the final step is where we submit all the data to the FDA or other regulatory agencies with all our evidences. Regulatory agency will take roughly about a year. If it's a normal submission, they will go through it. If they have any queries, they'll come back to us. EA will appoint a chair or a committee which will decide whether they can approve this drug or not.
For the metastasis studies, we have also used
emergency use authorization for which the COVID vaccine went through. That was done in a very fast time because it's an emergency authorization. It was done within a month. So we submitted the data, and then FDA reviewed everything in the next 15 to 20 days, and in the next 10 days, they formed a committee or a chair which approved the emergency use authorization of the COVID-19 vaccine.
Release analysis.
What is the significance of statistical analysis? So it's very critical for the correct interpretation of the result. It specifies how the outcome can be measured. So data comes in various forms, and it can be measured in two ways. Either binary, which is yes or no, and also what is the count of the particular adverse event, or as I mentioned earlier, it could be binary, whether yes or no. Whether it's a serious adverse event, yes or no. That would be the analysis, right? There are other measurements, right? In case if the measurements cannot be done by these two methods, either by binary or count, there'll be question. What we will get is basically like the answers for their questions, and then we have to create an analysis data sets analysis based on those questions. We have to decide what appropriate statistical tests that we should use and what is appropriate statistical procedures that we should use, like correlation coefficients, hazard ratios, etc., which makes sense and like which explains our data really well. We collect the data, we organize the data, we present the data in a presentable format so that the FDA can understand it, and then they can approve it faster. So what are the challenges of this clinical trial? Clinical trial has plenty of challenges. First thing is it's volunteer-based. As I mentioned before, I told you that a lot of time will be taken for recruiting the patients. That's because it's volunteer-based, and notice should be kept in many places to attract volunteers. Selection includes screening criteria, which are sometimes very complex. And then it will cost a lot of money. So the sponsor should be ready to shell out a lot of money initially without the guarantee that this drug will be approved by the regulatory authority in the future. And then the global regulatory process varies. So different countries have different regulatory authorities, and then the submission will be different. So yeah, these are all the challenges that we are facing in clinical trials. Now coming to AI. So talking about how AI is revolutionizing healthcare, first we need to understand what is AI and what are the differences between the names inside the AI, right? So AI is kind of the high-level term. Artificial intelligence says like any techniques that enables computers to mimic human behavior, right? That's right. So inside artificial intelligence, there is something called as machine learning. Machine learning has the ability to find patterns in data without being externally programmed, right? You don't need any programming. The machine has to learn everything by itself. And the third one is called as deep learning. It uses neural network models with many hidden layers. It is more advanced. So I would say it is a combination of both artificial intelligence and machine learning. Has AI chip in to accelerate the speed? Yes. It has really helped with the clinical trials and in general healthcare. It has accelerated the patient recruitment, which usually takes a lot of time. How? By introducing a simple app instead of posting notices like in different places. You just release an app and give an ad saying that download this app, and if you're interested, you can participate in this clinical trial and identify patients matching criteria in a short time for more complex trials. As the same app, you can load a set of questions inside the app, and based on the patient responses, you can decide whether this patient or subject would be eligible for this clinical trial. So it lowers labor cost related to maintaining of subject data. Help you to eliminate the third-party companies for collecting data. And then it can refer the subject patient to the trial visits when needed. Right? It also has given subjects or patients a cutting edge and medicines and therapies in a click. Teledoc is very popular. You don't have to visit a doctor. You just have to download the app, and then you can do a video conferencing with your doctor without visiting the clinic. The advantage is that you can get access to the doctor at any time of the day, even during weekends. Like the doctor doesn't have to visit the clinic. You don't have to go to the clinic to visit the doctor. It has definitely improved patient satisfaction scores. So talking about some of the apps, I will give you a couple of examples of how applications have been successful in clinical trials. MentorMate is a company which have developed an app called as Attached To. So Attached To, the explanation or the full form of Attached To is antihypertensive treatment in acute cerebral hemorrhage. Right? So what this app provides assistance with pre-screening, patient eligibility assessment, and randomization in a 5-year multi-center randomized controlled phase 3 trial. So using this app, they were able to recruit patients or subjects for the study much faster, and that the screening was done faster. So it basically doubled up the speed of the clinical trial. The second such app is called as AI Cure. So what AI Cure is specialized in, it visually confirms medication ingestion. It combines the power of artificial intelligence with deep learning, computer vision, machine learning, and predictive analytics. So it makes sure the right patient is taking the right medication at the right time. So this is another classic example of how AI has revolutionized the healthcare or clinical trials, right? So here in the picture, you see a conventional method for testing Parkinson's disease in subjects, right? This is called as UPDRS testing. It's called as United Parkinson's Disease Rating Scale. A subject, usually he has to go to a doctor, and then during the doctor visit, the doctor will tie a device to a patient, to the patient's hand, and then they will let the patient walk inside the clinic, and then based on their movement, they will calculate a score. Total score calculated by both the doctors during the patient subject visits, and also using the recordings of the patient. They have created a line graph which shows the effect of the treatment drugs against placebo. But to be honest, like this method is not so reliable because the patient sometimes can forget how strong the symptoms were, and there is a chance that they got a wrong symptom on a particular day. So these were the challenges in the conventional UPDRS testing. But now, so this is a study that was done by Pfizer's Innovation Research Lab, Boston University's Pivak Center for Clinical and Translational Neuroscience, and Tufts Medical Center's Department of Neurology. So what they have simplified this UPDRS testing by just providing a wearable, like kind of a smartwatch or a Fitbit to the patient, which they can wear almost all the time. So what this wearable does, it records the subject's motion as they walk. And then based on this data, they were able to score like pretty much what they did in the previous slide, what I showed in the previous slide, this line graph, they were able to do it. They were able to calculate the score, but they could calculate the score pretty much every day instead, like once in a while when the subject visited the doctor, like in the conventional method. So unlike that conventional method, this method is more reliable as the data is collected in real time and it can be calculated almost immediately. So using this variable, they were also able to look into some important data like PD patients spent less time walking than healthy controls. You could see that. They have also found out that the Parkinson's disease patients stand up or sit down very less often than healthy controls. And also that like Parkinson's disease patients while walking, they turn slower than healthy controls. So these are some of the very important data that they have collected, which were basically not possible to collect using the conventional method. Right. Apart from this, there are other AI softwares that have recently been approved by the FDA for diagnosis in clinical trials as well as in healthcare. First one is AI Doc MaxQ. So once we take a CT scan of a brain, it will basically help the radiologist to flag off the acute intracranial hemorrhage or bleeds in the brain. Right. So the second such software is IDXDR. It detects signs of diabetic retinopathy. So it automatically analyzes the images of the retina for signs of diabetic retinopathy. It has a very cool camera. It captures the images of our eye, and then the images are transferred to the IDXDR client software, and then the images will be analyzed using this the software's AI technology. Right. The third one is ICAT. Helps in detecting the breast density via mammography. Technology uses AI to assess breast density in order to identify patients that may experience reduced sensitive to digital mammography due to dense breast tissue. And then fourth is Biomedical. It measures the coronary calcium scoring. Coronary calcium scoring is a marker for coronary heart disease. Again, it needs a non-contrast chest CT, but it can automatically calculate the coronary calcium scores. The next AI software that recently got approved is Icometrics. So what Icometrics does is that it uses MRI images. It quantifies brain white matter abnormalities in patients, decreasing the amount of time taken, increasing the accuracy, and improving patients' care for those with brain issues. Last but not the least, the software that got approved very recently for stroke diagnosis is AI. It uses the CT scan and it automatically detects a large vessel occlusion and synchronizes care by alerting the related doctors. So these technologies, as I already mentioned, they are currently used in healthcare, but they are also used in clinical trials, thereby enhancing the quality of clinical trials and also by doubling up the speed. So this slide shows the usage of AI across various industries. As you can see, like about $150 billion, huge sum of money is invested in the market, which is dependent on AI. And about 60% of the top 10 industries are healthcare industries. So that tells you how much AI is influencing the healthcare industry in the US and across the world. Until this slide, we have seen like all the pros of AI. One of the important cons or concerns about AI is that lack of personal involvement, right? So interactions between a doctor and a patient are very important in building trust and treatment. No matter how good the AI is, no matter how good medication it recommends to the patients, I will not be comfortable of any robots or like any AI suggesting me medicine. I would be comfortable when I see a doctor, and when a doctor prescribes medication, I'll be comfortable with. The second concern is that usage of AI has caused a rise in unemployment rates among healthcare workers. So to give you an example, there are a lot of chatbots and robots that can provide mental health, analyze the condition of patient's health, and foresee some problems like seizures, sepsis, cardiac arrest, etc. Consequently, many people lose their work. The third concern is that there is a possibility of a defective diagnosis. Accurate diagnosis for a certain disease depends on the various data collected from millions of people who have experienced similar symptoms and conditions. So AI database has to be that robust, and it should have all the sufficient information in order to provide the correct diagnosis. If they don't have that many robust data, there's a possibility of defective diagnosis. And the fourth concern is social prejudice. So I'm not sure yet that the AI is capable of detecting the background of any human yet. But as you see, if you go and visit a doctor, the doctor will ask your race, the doctor will see your condition, and then appropriately he will prescribe you some medicine. But AI cannot do that. It cannot detect what background you are from. It may prescribe a very expensive treatment to a person who is not earning that much. So it's always a problem depending on AI for any treatment. So very recently, they have asked people whether they would be comfortable in AI helping them with prescribing medications or in healthcare, and these are the responses. So 47% people said they use AI because it is readily available. It is available in their cell phone. It is available using your laptop. And 36% of the people says that it saves time by avoiding a trip to the doctor. 24% of the people says that AI has a vast amount of information, relevant information, what they are looking for. On the other side, people say that they are not comfortable with AI because 29% of the people say that they like visiting the doctor. 26% feel that they don't understand the AI and all the other apps that AI uses. And 23% of the people say that they don't like to share the data. I'm very surprised that like 23% say that they are not comfortable sharing the data. I was expecting more than 50% to say this reason. In summary, AI has definitely revolutionized the healthcare. So it has doubled the speed at which the diagnosis can be made and treatment can be given. More innovative things are in the pipeline in the foreseeable future, which will definitely revolutionize or reform the healthcare industry. So is it boon or bane? That question still not been answered. We have seen both the pros and cons of the AI. Only time can tell whether it will be a benefit or not to our healthcare industry and in general to our society. So as I already mentioned, I work in Pfizer. This is our New Jersey campus. They do have campuses across the US. The headquarters is in New York City, which is an hour drive from this campus. But this is just a beautiful campus. I took this picture from the site brochure. I guess they have taken this picture during the fall time where the trees will show different colors. As you can see, like it is surrounded by hills and a lot of greens. It's a nice place to work. Thank you, and I'll be happy to take any questions you have.
So, hello everyone. Vanakkam. I'm Professor Dr. Mandina Shantaram, Department of Biochemistry, Mangalore University, Mangalagangotri, Karnataka, India. And first of all, I thank the organizing secretary, Dr. Padmini, for having given me this golden opportunity to participate in this international webinar. Now I'm going to speak on anti-Alzheimer's activity of plant isolated compound, APPL Garifol Drosophila melanogaster model. This work was performed by Mr. Pratap GK, one of my PhD scholars. He has submitted his PhD thesis in November 2020 and is waiting for his final viva. Well, as we all know, medicinal plants are potential renewable natural resources considered to have a beneficial role in human well-being. Natural products and their derivatives represent more than 40% of all the drugs in current therapeutics. Because of the high success rate and less investment need, today's synthetic drugs are very expensive and they have considerable side effects. Alzheimer's disease is one of the most common age-related irreversible progressive neurodegenerative diseases. The main characteristic feature of Alzheimer's disease is the gradual decline of a person's memory, ability to learn, reason, and difficulty to carry out daily activities due to the accumulation of cortical plaques in the neurons. Medicinal plants are the most effective sources and drug development, and the majority of the plant products that have been derived from them are already at the clinical trial stage. Due to this lack of conventional therapy in recovery of neural damage with less side effect and with the harmful side effects associated with administration of synthetic drugs, there is a crucial need to look for substitute treatments in the form of herbal drugs or medicinal plant products to cure such AD diseases. Indigo is a small herb found in India throughout the Western Ghats of Karnataka, India. It belongs to the family You can see the on the slide of Curligo orchioids gagging. Pharmacological studies showed that orchioids are used in several disorders as antimicrobial, anti-tumor, hepatoprotective, antioxidant, immunomodulatory, cardiovascular activity, ischemia activity, ischemic activity, anti-diabetic activity. Based on the literature survey, no one identified the bioactive compounds from fertilized plant source. Now, MS1 compound, which was isolated from Curligo orchioids, excellent acetylcholinesterase activity, inhibition activity in vitro and in vivo studies like Ellman's bioautographic method and also in transgenic Drosophila melanogaster. And as I have already mentioned, Curligo orchioids is commonly known as golden eye grass, and its common names in Kannada are Nelatale, Nelatengu. And based on materials and methods, these plants were collected, identified, and the compounds were extracted, and biological activity of food extracts were performed by our group, research group. Isolated compound was analyzed by LCMS, HPLC, FTIR, and CNH NMR. Then APPL GAL4 Drosophila strains, and the GAL4 UAS system is a biochemical method used to suppression and function in organisms such as the fruit fly. And thankfully, APPL GAL4 strains were generous gift by Dr. S. Sri Krishna, Professor, Department of Biochemistry, Institute of Science, Banaras Hindu University, Varanasi, Uttar Pradesh, India. And the experiment, we started the experiment with the MS1 bioactive compound to be used. And the experimental diets containing MS1 bioactive compound was prepared. And in vivo neurotoxic study was done as per the method of Haddadi. And prior to exposure to MS1 bioactive compound, flies were fed with the cream agar diet, which comprises control group, and the second group fed with diet containing MS1 compound with various concentrations, starting from 0.02% to 0.06% weight by volume, dissolved in 0.5% DMSO, dimethyl sulfoxide. Then once in every 3 days, after the flies were transferred into fresh media for up to 30 days. After 30 days, the heads of 50 flies were homogenized. And the lifespan assay was carried out according to the protocol explained by Finelli. And reactive oxygen species assay, lipid peroxidation assay, catalase activity, and acetylcholinesterase activity were determined by all the standard methods. Even histopathological studies were conducted. And to say in a very briefly, the flies were anesthetized, fixed in Tournois fixity at 4 degrees Celsius overnight. After the fixation of fly heads, the samples were dehydrated in alcohol, and then fly heads were embedded in paraffin wax. Sections of 4 millimicron thick heads were stained with hematoxylin eosin stain. Even the climbing assay was performed. Locomotor activity and the climbing assay was described. It was performed as per the description of Lee with a slight modification. Molecular docking studies on MS1 compound was also carried out. Molecular docking to gain insight into the molecular determinants that modulate the inhibitory activity and the mechanism of inhibition. Modeling study was performed to explore the binding interactions of MS1 compound to the Drosophila melanogaster acetylcholinesterase, human acetylcholinesterase, and beta secretase enzyme. These enzyme molecular docking trials were performed using the AutoDock tools 1.5.6. And as per the table, it was found, you can see that it was found that MS1 compound had the least free energy of binding minus 7.45 kilocals per mole, closer to the standard drug galantamine. And in the next table, we can see MS1 compound binding energy is comparable with the other standard drugs of studied by other scientists. ACH, that is acetylcholinesterase and beta secretase 1 important enzymes, which play a significant role in the signaling pathways of several Alzheimer's diseases. Discovery and inhibitor design of ACHE and beta secretase are of therapeutic importance. ACHE and beta secretase enzymes have recently emerged as a key target in drug discovery. It has been implicated in multiple cellular processes and linked with the pathogenesis of several diseases. ACHE and BACE enzyme inhibitors might show valuable as therapeutic natural compounds in the treatment of conditions linked with enzyme activity such as Alzheimer's disease. Now, as you can see, the diagrammatic representation here, when MS1 compound was used on AD model, APTL GAN4, it was found that oxidative stress level was decreased, climbing activity and lifespan were increased, ACHE and beta secretase activity were decreased by MS1 compound. And when we come to results and discussion, we have found that MS1 compound is a triterpenoid compound. Now, looking in all these four graphs, we can observe that supplementation of higher concentration of MS1 compound has decreased the oxidative stress markers such as reactive oxygen species, catalase activity, and lipid peroxidation, as well as acetylcholinesterase activity. Now, next two graphs. Here, supplementation of higher concentration of MS1 compound has increased the lifespan of Alzheimer's disease Drosophila fly models and also their climbing activity. Now, when you see the diagrams of the control eye and Alzheimer's model of a fly, as per this slide. In this slide, we can observe the control eye in wild-type Drosophila intact eye morphology, no loss of corneal lenses, that is ommotidea, there is no damage at all. That is shown in the A labeled pictures. APPL GAL4 model, in this, loss of eye morphology and corneal lenses are observed. That is B labeled pictures. In C and D pictures, MS1 compound treatment exhibits reduction in loss of eye morphology and corneal lenses. Higher the concentration of MS1 compound, minimal is the disruption in eye morphology that was observed. Now to summarize, herbal drug preparation from a whole plant or parts of plants, bark, leaf, root, rhizome, and so on, and development into a plant food drug was the concept considered in our study. It is useful for the pharmaceutical formulation. Traditional medicine that involves a broad use of medicinal herbs and medicinal herbal drug preparations that is known to treat different neurological disorders like Alzheimer's disease and Parkinson's disease, etc. MS1 compounds as a good anti-ACHE agent, anti-amyloid activity in vivo model of transgenic APPL GAL4 Drosophila model. The current study has created only a baseline data as a scientific evidence regarding the beneficial effects of plant compounds and plant extracts used as antioxidant and effective inhibitors of anti-acetylcholinesterase activity and also neuroprotection. To conclude, the obtained results might be useful in explaining the effect of MS1 compounds as a good anti-ACHE agent, anti-amyloid activity in vivo model of transgenic APPL GAL4 Drosophila model, and its ability to protect the neurons against amyloid beta, and in future it will be used for anti-Alzheimer's disease drugs for the management of Alzheimer's disease. Now, just a few recommendations for future studies. Study has created only a baseline data as a scientific evidence regarding the beneficial effects of natural compounds and plant extracts used as antioxidant and anti-ACHE activity and in vivo model of transgenic APPL GAL4 Drosophila model. However, extensive research in various directions is required to develop natural products or plant-based products. Results are reliable and more studies are necessary to further verify the anti-AD effects of MS1 compounds in vivo studies using transgenic animal model like AD tau rat, APPL21, and APP31 transgenic rats, APP23 rat, APP23 mouse, Pinman knockout mice, then Cynorhabditis elegans and zebrafish model of OKA-induced AD can be taken up for further studies. These are some of the references, and thank you very much for your patient hearing. Thank you.
Good morning to all the participants who have joined this IABMS conference today. It gives me immense pleasure to share some of my research ideas with the elite audience and dear students. So the reason for this presentation is that let everyone you encounter today live happier and better than they were before. So we'll go to my topic. Today, I have chosen a topic which is very interesting and dear to me so that I can share with you. Applying machine learning methods in deep phenotyping during pregnancy-induced hypertension and preeclampsia. The contents of the lecture is going to be this way: pregnancy-induced hypertension, preeclampsia, artificial intelligence and machine learning, deep phenotyping, applications of machine learning and deep phenotyping, and more information for the aspirants, more information about AI. So first, I thought I'll give you some basic information about preeclampsia. Preeclampsia is a pregnancy-induced disorder. You see this picture, the outcome of this disorder is felt globally every year. In 2015, the statistic says that nearly 70,000 women, they died because of preeclampsia, and 5 lakh babies, okay, all over the world, they die because of preeclampsia. That is the magnitude of the disease that we are going to talk about. And if you see the global publications, more publications for preeclampsia either comes from USA or India. That means 25% of the publication has come from US, and at least 16 to 17% of publication comes from India. That means that India is having at least 8 to 10% of pregnancies that is affected by preeclampsia. So this was the interest we had, and we started this study in 2014 with a team of wonderful research scholars with me, where I started with placenta first. I took the placenta from the normal pregnant women and from the preeclamptic pregnant women, and we were able to do basic analysis to find out the abnormalities, that is biochemical abnormalities between the placenta and the placenta of the normal pregnant women and the preeclamptic women. And from there, we started moving to study about the trophoblast, placental trophoblast, which are very important during pregnancy, because the whole placenta, the whole process of pregnancy begins only with trophoblast. There are 4 types of trophoblast. We work with all the trophoblasts. And then this trophoblast will eventually displace the endothelial cells. So we started studying about the endothelial cells, and we collected a volume of information. From endothelial cells, the next important problem during preeclampsia is infections. Infections that are caused by various microorganisms. We took up urea plasma urolithecum, a common cell microorganism, and that we imposed in the placental explants, and we did a series of biochemical analysis. Finally, we did placental explant analysis. So this was what we started from for the past 10 years, from 2014, 4 to 14, we were doing these experiments. And the outcome of the experiment very clearly states that pregnancy is a situation, normally pregnancy is a situation which has a stress condition. But when it comes to preeclampsia, there is an enormous change in the placenta in terms of the heat shock proteins, in terms of all signaling molecules, and then even the basic enzymes that are necessary for proper placentation also varies from normal to preeclampsia. This is the information we got, and then from there, okay, we have studied all the important aspects of preeclampsia, but where is the therapeutic intervention? Where is that you can find a remedy for at least managing comfortably the preeclamptic situation? Then we started with the herbal preparations, profusely funded by the National Tea Research Foundation. We used black tea, we used green tea, then we used mint tea. We also used betel leaf extracts, then we used squid, then canberry extracts. And all these extracts, when they were incubated with the cells or the explants, they gave very good results in such a way that these incubations can balance the deformity caused due to preeclampsia. But all this is only in the in vitro condition. So if it is in vitro condition, in order to validate that, we have to go for clinical studies. But The clinical studies are not possible because if the clinical study we have to do, it should be a cohort study, it should be along with the hospitals, and they should be follow-up, and so many other constraints are there. So I started looking out for any other process by which we can have a remedy for this. So as I was going through in the literature for further work, apart from that, before that, I'll tell you the publications we have. Just show the publications. What we did was, with this information that we collected for 10 years, 70 international papers were published. And these till then also, till 2020, also there was no treatment for, clear-cut treatment for preeclampsia. Even after we did the herbal, it is not considered as a treatment, it is only a management technique. Then, when we looked into the literature, we found out that in 2018, there are some possibilities, because that is the time when CT scans and our MRI, all those became very popular, or they became advanced. And we saw that these kind of imaging techniques can have some solution where we will be able to identify the preeclampsia much earlier, so we can have some conclusive management technique to prevent death for the mother and the fetus. So in the process, we published all these papers. These papers are very valid papers because they are the basic data that is given for the artificial intelligence that we are going to talk today. So the next slide will now show you about what is pregnancy-induced hypertension. So the pregnancy-induced hypertension can be broadly classified as chronic hypertension, and then it can be classified as pregnancy-induced hypertension. Chronic hypertension are not bothered because those conditions occur when already the patient, the mother had the hypertension, and then it continues that way. But hypertension during pregnancy is only after becoming pregnant, the mother shows a sign of hypertension. So that is again classified into three categories. One is gestational hypertension, the other one is the preeclampsia, and then it's eclampsia. Now, what's the difference between gestational hypertension and preeclampsia is, gestational hypertension is there's an increase in hypertension from 120 to 140/90, but no proteinuria. But if it is a preeclampsia, then there is a increase in blood pressure above 140. It can be 140, it can be even 160. And then again, the blood pressure I'm talking about. Then the proteinuria level is certainly found, that is about 300 milligrams in 24-hour urine sample. So this preeclampsia again is further classified as mild preeclampsia and severe preeclampsia based on the other conditions, like somebody can have a seizure. If they show seizure, they show a headache, blurred vision, then that is classified under severe preeclampsia, and that is about more than 160. Their BP will be more than 160. So what we worked always was only with the mild preeclampsia. So this is the overall look about the pregnancy-induced hypertension and preeclampsia. Now you have to really understand what is preeclampsia. Preeclampsia is a multi-system disorder of unknown etiology, and there is a development of hypertension to the extent of 140/90, and then again you have proteinuria. All this is expressed only after 20 weeks of gestation. This is the important point you should know. Then if you consequently see the classification I've already given you, if you see the symptoms, I'll slightly make it more clear for you. See the symptoms. You must keep this in mind, it's very important. So it is 140 above becomes hypertension. 300 milligrams in 24 hours urine is your dipstick method. You can find a proteinuria, and you have edema in the hand and face. So this is a definite symptom which appears only after the 20th week. So in normal pregnancy, what happens? You should understand normal pregnancy, and only you can accept the preeclamptic situation. In normal pregnancy, you will see that there is a fetal trophoblast which will invade the walls of the spiral arteries. This invasion is very prompt, and this disturbs the smooth muscle layer, that the endothelial cells that is present in that layer, and then it will convert into venous-like channels. Now, this remodeling begins at the 5th or the 6th week of pregnancy. This is a very important point you should remember. And it continues to 20 to 22 weeks. Now, why should all this happen? Because the blood supply to the fetus, from 10 to 50 ml, it should go to nearly 800 ml. so that there is a proper development of the fetus. This is what happens in the normal pregnancy. Now, what happens in pre-eclamptic condition? There are few arteries that are undergoing change. That means when the trophoblast invasion is abruptly not functioning properly, so endothelial displacement does not take place. So there is a problem. More than that, these changes does not happen throughout the myometrium in spiral artery. So the spiral artery formation itself is defective. That means the blood flow is incidentally not proper. There is a problem in the volume of the placenta. There is a problem in the development of the fetus. So now you see, you must also understand the pathology of preeclampsia. The preeclampsia shows there's a two theory. One is a vascular theory and other one is alloimmune theory. Now the vascular theory profusely talks about the maternal microvascular diseases that includes the abnormal placentation. Now if you see the alloimmune theory, there that theory is talking more about the immune system and also the sperm exposure and other things. So these are two categories. We are basically working only on the vascular theory. So to sum up, I'll give you the pathophysiology. This is a systematic inflammatory response that is due to the endothelial action and maternal symptoms arise. The second one is where you have the generalized edema situation, translative edema. Then you have vascular damage, which I'll be talking about in a few minutes from now. And then there is a vasospasm. So this is all that you have to remember, the pathophysiology. It's a really serious one, if you will understand. Then what are the other complications that will happen for the mother and the fetus? Now you'll see this chart. If you see this chart, you'll see the mother is first, she develops seizures. There's a problem at the brain level, at the hepatic level, at the renal level, at the lungs level. So everywhere there is a problem due to preeclampsia. It gives complications. So also for the child. The child, definitely preterm delivery is a hallmark of preeclampsia. Then after which, even after the delivery, there can be a problem. So that is the actual story about the preeclampsia. Now, when you come to the treatment part of it, the treatment part of it is that there is no curative therapy for preeclampsia. The delivery, the only way where the placenta comes out of the body. So because it is placental induced disease, there is a possibility that that particular discomfort for the mother is removed. But I told you in a previous slide that the mother has other difficulties. So what is the kind of treatment they are doing? If it is a mild preeclampsia, they will be giving these medicines, magnesium sulfate. If it is a seizure and other things, they change it a little bit and give some other type of medicines. And that's all it is, just to keep the mother in rest and just to see that there is no loss of blood and the pressure does not rise and all that. But that is not a comfort pregnancy at all, and still the outcome of the pregnancy is not sure. So that is why I showed you that 76,000 deaths, 5 lakh children dying happens. So that was the condition. That is the condition even till today. But then, as we were looking into the literature for some more information on preeclampsia, we found that the artificial intelligence studies can definitely give a predictive analysis of this preeclampsia, and from where there will be a ray of hope for planning the pregnancy for the mother. So what is artificial intelligence? The artificial intelligence is nothing but a computer science technology where the computer is made to understand a problem. It is made to take decision on a problem, and the computer can also give you a solution based on what is the input given. Under the artificial intelligence, you have what is called as a machine learning. So artificial intelligence is like the syllabus. Machine learning is like examination. The machine has to learn how to use all these information given to the computer, and then using these statistical techniques, and then it has to experience the intelligence that it has given to you. From there, it again goes to the deep learning technique. Now, the deep learning technique is a subset of the machine learning, where the neural network, it's called it's just like the brain. The brain has neural network. Similarly, deep learning techniques also use neural networks, and this we're using appropriate algorithms, and then find out a problem Once you give an input, it gives you an intelligent output. So this technology started 20 years ago, but then this methodologies, they were applying after the advent of genome mapping, I think from 2010. So artificial intelligence and machine learning methodologies naturally apply to characterize phenotype in a deep manner, including imaging, bioinformatics, genetics, diagnosis, and so forth. So the next thing is that these methods can exhaustively examine the data with high granularity and dimensionality, and may make use of the broad range of data types that may be processed by phenotyping. Now, several disease outcomes during pregnancy is complicated, just like I showed you now, and it is difficult to understand the etiology, leaving very little that can be done for prevention. So we cannot do the prevention. We don't know how to do the prevention. So what next can you do? If you cannot prevent, can you detect it much earlier? The next question is, so deep phenotyping these patients can help us to detect the disease in a much earlier stage and do the needful. So now the question is, how is it that the computers are able to do this? Now, if you compare the neuron in the brain is 10 to the power of 12 neurons we have, and the cycle for it is only 1 millisecond. But if you take a super good computer, it has 10 to the power of 8 or more transistors per CPU, and it can have hundreds of CPUs, and it can have 10 to the power of 12 bits of RAM. That means the cycle time becomes nanoseconds. So that means you can see the speed with which it can see the data and give you the results. So naturally, you have to believe the computer, if you train the machine properly, it can give you very useful results. Then from there, we go to about one classical example. See, you see here, you have an input. Usually they term it as K. A small portion of that K is taken up, so convolutions, that is called, or image mapping. You see 3 mapping. From the 3 maps, they take a small bit, a sub matter in that, and that is expanded to about 12 units. And then, like, for example, this is a simple example, then that is again multiplied multiple times, and using neural networks, you see the output. So this is a simple basic understanding. You just take one small bit, expand it to the maximum level possible, and then you get an output. For all that, you have to have a mathematical, biostatistical, and algorithm designing. So that is the example I showed you. So with this technology, what we can do today is, today's healthcare system can be very easily handled with this kind of technology. So that is what this figure shows. For that, what you need is, you need digitalized clinical data in an electronic health record that is available in the hospitals. Genomics, that's already been done. We can always do it in genomic laboratories, and biology. That is where we all come. So all biologists, they have to work, they have to generate data, and they have to publish. These data are very, very useful for healthcare systems. That is why I showed you earlier, we have given, very proud to say that our students, they have given 70 and more good publications to the mechanisms how preeclampsia takes place. So the biological input is very, very important. So this wealth of information is very important, and artificial intelligence and informatic methodologies can be applied. Then learning and fine-tuning healthcare system happens. From there, from this point, you can have the outcome. And the outcome is going to be deep phenotyping that we are going to talk about in a few minutes. So if you see, disease phenotype refers to a deviation from healthy morphology or a physiology. So the study of the phenotypes require a spectrum of phenotypes associated with the disease entity. Only if you have the data, we can do that. So a healthy morphology or physiology, first of all, you should know data. What constitutes the disease phenotype, we should know. Then you can do deep phenotyping using those data. And that is the step towards precision identification of the disease at a much earlier level, which will give you what you call an advancement in the disease presentation and prevention. So I'll show you this. So first I talked about the data. See, you see all the data that you publish in PubMed or Scopus, these records. So for example, if you have 525 records, they have collected good records. These records, first of all, they are corrected, then they undergo a screening process. See, they undergo a screening process. When they do the screening process, again, they have the records being shortlisted. Then you have the eligibility. Around 200 records are available. Then other records are excluded, and they finally end up with the small records which they can use for whatever is the aim of the method for which they are going to use. So that is where the data collection takes place. Apart from that, now you have to see the methodologies, what you require for the inputs. See, if you see the input for medical information, you need pregnancy-related processes, you need fetal state, you need birth defects, gestational dampness, preterm birth, and all this process required as a medical information. This is very, very important. Not one, but the data should be very big. That means you should have at least, say, for example, 5 to 10 lakhs pregnant women's medical information should be there. Then, if you go to the artificial intelligence and IT information, you will have artificial intelligence topics where you have the data acquisition, methodology, and knowledge representation. So this is what is methodology and knowledge representation is based only on the medical information. Then you have to do the data preparation accordingly, then prepare accordingly the algorithms, then model validation, then performance metrics, then framework and programming the languages. You should have very good graphical user interface, and also you should have IT security. With all this, you can go ahead and do the analysis. So I thought I'll just give you a broad introduction to machine learning techniques. It is broadly classified into 5 types. You have the supervised learning, unsupervised learning, and so on and so forth. And then you have the deep learning. So deep learning is what we are going to focus. All these informations you would have already seen during the introduction we are going to have from Professor Jyoti. So I'm going to talk only about the deep learning technique where neural network is used. So where it is used is the sample here. You have this data. You have the myriad data types. See, one is in genomic studies, and you have other omic studies. You have imaging systems, phenotyping, exposure systems, and then you have the clinical studies. So all the data from where we get data to do the studies. So far, there's a successfully the artificial intelligence and deep phenotyping has been applied. The classical examples are diabetic retinopathy. You see this, the disease state. There is you can predict the disease state here with this picture, and how far is it diabetic retinopathy. Then similarly, the mitotic cell count in the breast cancer has been identified. So you do staining for the breast cancers or any cancer, that can be read by using this deep phenotyping. In case of Alzheimer's disease, you can identify using phenotyping. The heart volume to find out in the case of coronary artery disease, that can be identified. They are using for protein studies. So these are all successful study outcomes. Now in future, they are planning to predict the toxicity of the new drugs. Actually, we have one session, one scientist from Pfizer is also talking about clinical trials, wherein he will be giving details. I think he'll be giving details about predicting the toxicity of new drugs. And then you can also understand gene mutation and prevent diseases. They've already started working about this in Toronto University. So I'm going to give you only 2 examples. Okay, the 2 examples, one is the placental volume and the other one is the residual vasculopathy. So fully automated real-time 3D ultrasound segment to estimate the first trimester placental volume using deep learning is available today. That is one. That is the one I'm going to explain to you. The second one is residual vasculopathy and its identification in whole slide image using multi-resolution hierarchical convolution networks are available. So these 2 techniques are going to give you 2 things. You can find out and during the pregnancy, you can find out whether this person is going to end up with preeclampsia or not using the 3D ultrasound technique. The second one is by studying the residual vasculopathy, you can surely tell that this person is going to have these problems during the next pregnancy. So let me explain, and in the end of the explanation, you will agree with me. So medical imaging technique is very important for the physicians to visualize the inner anatomy of the patients for diagnosis and analysis purpose. Now you know about MRI, CT, and ultrasound imaging. This is all very popular today. So this ultrasound technique, we are very much interested because it is, first of all, economical. And ultrasound, what we have today is the 2D ultrasound. Now this 2D ultrasound using the deep phenotyping and machine learning technique can be converted into 3D visual technique, visualization, wherein they use VNN, DW, and so on, all those algorithms. So this is what is going to be very useful for us as far as the ultrasound is concerned. And now, the 3D ultrasound imaging system visualizes the region of interest. ROI is called as a region of interest. See, whenever you take a step, there is no pathologist who can be seeing everything, every small detail about a particular problem. So they have what is called as the ROI. So the machine will be able to detect the region of interest and then convert it into a 3D system. So that is what this graph shows you. A 3D ultrasound imaging system will have a 2D array scanning system, either tilting, linear, or cylindrical. You can do this tracking this way. This is how the probe is being used in the freehand system. So once you get the data is acquired, and this data goes to the next step, how it is being used. The next step of the data acquisition, you have the next step. The data is being processed by the machine. After processing, the volume construction takes place. All that it processes, it will not use. It will do a reconstruction, and then finally it gives you a display or a visualization. This is what it's using. So if you see this picture, you are seeing here, this is a 2D array. From one point, you will be able to protect and see so much of details. The details to this extent, you can get a picture. See, there's only one point from here. This is called pyramidal system. 2D array into the 3D scan. So from one point, you see nearly you can see 6 into 6, 36 points. So that kind of depth in the visualization. This is the 3D system. This is the 2D system. So this is the mechanical system the artificial intelligence is giving you. So these are all three types. So one is the surface rendering technique, then multiplanar technique, and then volume rendering technique. All these three techniques are used to give you the ultimate result. So the result here shows that, see, this is the surface rendering technique. This is the, I didn't get for the placenta. So for example, I'm showing the heart. See, this is the heart if in a normal 2D scan, and this is what happens in the 3D scan. You can see all the details. This is called as a 3D rendering technique. Now, this is interesting information that I want to share with you. Now, get back. Yeah, so this is a very interesting information. This is the placental picture with a multiplanar reforming technique. This actually shows the volume of the placenta. See, there is one thing in the system called the small for gestational age. It's called small for gestational age birth weight. That means the placental volume you can measure in the, say, 6th week of the pregnancy, and they can check the volume of the placenta. And the volume of the placenta should be appropriate with the weeks of gestation. If the volume of the placenta is less, that shows that already there is a problem in the spiral arteries. There is no proper blood supply, and that is why it is called as small for gestational age. So that means the child is not able to expand and grow. Already fetal restriction, growth restriction is started at the 6th week. So this gives you, that is placental volume, clearly gives you the idea that this person will end up in preeclampsia. The next one is the multiplanar reform technique you see here. That if you see that picture, in that picture, the artery size itself is see, you can see the normal one and you can see the abnormal one. The size is different, and by doing this studies also, you can see that the remodeling is not done properly. Now, if you see this, it's a graphical representation of actual placental volume. And see here this picture of the fetus, how it grows, the restriction is there. And this is a 3D picture, and this is the arteries. This is in low resolution, this is in the high resolution. So this kind of output is possible in the 6th week of pregnancy. That means at the 6th week of pregnancy, you will be able to define that the mother is in the process of developing hypertension, and there is a problem in the process of remodeling. So the doctors can take decision at that point and see, for example, give any type of drugs that can help in the management of the preeclampsia. Today, what they're doing this management after the 22nd week, when already there is proteinuria in the urine and already the edema has started. So if you can start at the 6th week and do the proper management or therapeutic interventions accordingly, you will be able to prevent the outcome of the disease. That is the idea. So that is how this scanning, 2D scanning to 3D scanning done by the machine is helping us. Next slide. So the next one I'm going to talk about is that DV. ML framework introduced a hierarchical method to analyze the histological digital images for the purpose of automating placental DV lesion inspections. So if you see that, the algorithm ability to discriminate the key feature and the candidate locations within a high resolution whole side image, WSI is a whole slide image, has been demonstrated, and this gives you this while keeping the false positive minimized. So if you see what happens in all these cases is the vasculopathy can easily be detected. Residual vasculopathy. When do you detect this? After the pregnancy, the plasma is thrown out. You can take slices of the plasma and use the plasma for pathological slide. You can make a whole slide, image slide with that. But if you give it to a pathologist, it is not going to be possible for them to analyze the entire whole slide. So, whereas if you teach the machine to analyze this, it is possible that immediately after the delivery, the machine will analyze the pathological samples and give you the result. That is what we are saying here, that when this risk of preeclampsia is identified earlier, it allows preventing treatments such as low-dose aspirin to delay the progression of the disease during pregnancy. When? For the next pregnancy. That's what it means. So accordingly, you can reduce health degradation for the fetus. So this is a second classical example where I'm saying you can use the deep phenotyping, take the placental sample, do the whole slide imaging, and give it to the computer, do the deep phenotyping studies, and get results where you will know whether there has been any placental DV. Okay, residual vasculopathy, if it is there or not. Once it is there, accordingly, you can do the treatment. So I'll show you this. So if you take a whole slide like this, and this whole slide, you can do 3D scanning. After doing the 3D scanning, by doing deep phenotyping, you can reduce the noise and find out the tumor. This is a tumor slide, okay? You can find out the tumor at this point. So normally it is not possible for the pathologist to that extent to study this, but it is possible by deep learning techniques. This is a classical example, but this is for a cancer cell. Similarly, if you see this MR imaging, they take the MR image, you have the input layer, then you have convoluted layers. From layer after layer, it will be pooled and featured and repooled, and then it will be vectorized to such a small extent. And from the vector, they'll be getting an output diagnosis that shows there is a deficiency in this particular region. So this is again done by the deep screening technology. So this is what I was talking about, the whole slide. See, this is the whole slide pyramidal approach. And from the whole slide, now you have digital screens which takes an entire picture of the slide, and then it is going to the computer network. So once it goes to the network, the subnet will be able to identify these components in much minute details. From there, again, they will reduce the physical size, and slowly they will reduce to such level that it will be to the power of 128. From 128, 1 into 128, and then 10 into 128. Then again, it will become 1 into 128. Sorry, this is 100 into 128. And then this you have 1 into 5. They reduce it to such level, and finally you come to the singular level to find out whether there is a DV. The patients, this is possible by doing deep phenotyping. This process is only called as deep phenotyping. So detection of pathologic placental blood vessel lesions, residual vasculopathy has been shown to predict adverse pregnancy outcomes such as preeclampsia, which can lead to mother and neonatal morbidity in subsequent pregnancies. Once detected, preeclampsia can be treated, so there is a considerable benefit from identifying at-risk mothers before symptoms can appear. However, although there are hundreds of blood vessels in a single slide, only one diseased vessel is needed to indicate the risk. This type of artificial intelligence approach can allow many more placentas to be screened with fewer pathologists, increasing the DB detection for mothers who are at risk for preeclampsia in subsequent pregnancies. This can accordingly reduce the healthcare expenses and reduce both mother and neonatal morbidity, particularly in the developing world, where perinatal pathologist experts are not really available. So again, I would come back to show this, to emphasize this point. This is, this is a slide used hematoxylin and eosin. This is a whole plasma cross section, and this is the normal one, and this is the preeclamptic one. And when you go for deep learning, this slide has got so many spots to identify DB, and from there they confirm that there is a DB. So the precision is more. Here the precision obviously is less. In the next slide, so what are the other applications? So, okay, can you do only for preeclampsia? That's not the case. There are several other interesting informations that I would like to share for the student. They are doing segmentation analysis using the deep learning image analysis. They are doing localization analysis. Each one is a specialization. Then you have quantification and diagnosis using the computer. Each one is a specialization, and you can learn any one of this. You can go for segmentation, means it has a set of information that you have to localize, you have to quantify, then you have to have a computer-aided output. So this is what you do. So we have ultimately come to the summary. So achieving adverse pregnancy outcomes can bring enormous loss to both family and society. We all know that. So pregnancy outcome prediction is a crucial research which will help reducing birth defect and improving the quality of population. So applying machine learning techniques, one can learn deep learning algorithms along with the available medical records and research inputs. This will detect and clarify adverse pregnancy outcomes even before the parent gets pregnant, during pregnancy, after pregnancy, and about the next pregnancy. That is what we discussed so far. So in conclusion, development of novel informatics methods focused on improving pregnancy outcomes remains an active area of research. So all our students should try to put yourself into this active area of research. The purpose of this talk is to systematically highlight the way that artificial intelligence and machine learning, including deep learning methodologies, can inform patient care during pregnancy and improve outcomes. It can be applied for other diseases. At present, ML and AI methods are being employed in selected units to optimize pregnancy outcomes, including modern DL methods. Future research should focus on the pregnancy domain areas, including postnatal and postpartum care. Also, clinical adaptations and implications can also be studied. So, what is the how will I conclude this the lecture? See, you see, this is a comprehensive slide. How pregnancy, the machine learning is useful for pregnancy. See, you see, first of all, counseling is done. Some amount of it is being applied for counseling. Then you see in vitro fertilization, machine learning is being used. Now, if you see ultrasound, 2D and 3D scanning, it is being vastly used. Then for the neonatal care, then for delivery, and finally also for the outcome and the records. So everywhere already artificial intelligence has taken its place for the care of the mother and the child. So women and reproductive health is very, very important all over the world, and artificial intelligence and machine learning has already given its best output. So the take-home points are artificial intelligence and machine learning methods can be successfully employed to optimize pregnancy outcomes. Today, supervised methods are the most common approach, and deep learning methods are gaining traction. Finally, future research should focus on less studied areas: maternal health needs, postpartum care, pregnancy care for transgender individuals, and in algorithms improvement, refinement, and translation of results back to the clinical care. So what are the other applications? Also, you should know, this is used for the gene expression analysis, medical imaging and analysis that we saw, cell nuclei segmentation, Then gesture recognition that is used for Parkinson's and other disease. You can do RNA, DNA structure predictions. You can do protein domain detections. And also I told you, you can use it for new drugs invention. So you have so much, all the students who are watching this, you have so many areas where you can put your expertise and work. So if I have impressed you, that is the happy day for me. So thank you very much for your patient listening. And what I want to really conclude is that there is no reason and why that the human, if you see the statement, I don't want to elaborate on the statement, but what I can tell now is that artificial intelligence has to go to its full dimension for us to understand. Thank you very much.
Knowledge about polypharmacy among pharmacy students. This is the abstract. And the keywords for this abstract is polypharmacy, side effects, interactions, and areas. Polypharmacy implies prescription of too many medications for an individual patient with an associated higher risk of adverse drug reaction and interaction. It is a preventable problem. Physicians and clinical pharmacists have the potential to combating this problem through a variety of interventions such as reducing the number of medications taken, reducing the number of doses taken, increasing patient adherence, preventing ADAs, improving patient quality of life and drug cost. The aim is to assess the knowledge about polypharmacy among pharmacy students. The objective is to develop a brief instrument for academic pharmacists to use the knowledge of polypharmacy. The main objective is to reduce the risk of side effects and interaction. This is a community-based cross-sectional study using questionnaires. The entire study was planned to be carried out for a period of 6 months. This is a cross-sectional study which involves pharmacy students. And the limitation of the study is the study could have been more generalized if it was multi-center involving other pharmacy colleges also. The limitation of this survey-based study is its small sample size. However, such study involving large sample size can be carried out. The main limitation of this study is that data collected were self-reported, which may introduce some bias in the behavior pattern of the respondents. Expected outcome: the pharmacist plays a key role in the practice of de-prescribing and must be practice ready in order to address the complex needs of an aging population. It has been suggested that a key change needed to fulfill this role is instruction on de-prescribing in pharmacy education. This exploratory study assessed if students were receiving instruction on de-prescribing and the format of the instruction. The results contribute to the discussion regarding de-prescribing education and can be used when developing the curriculum and activities on the topic. Thank you.
Hello everyone, my name is Sanjana Arun from Wales University and my abstract ID is 596 and my title is on knowledge, attitude and practice on the use of self-medication among general population in Chennai during the lockdown 2020. Abstract. During the coronavirus pandemic, which occurred in the year 2019, where millions of people have been affected from this disease worldwide. Irrespective of the lockdown, which was issued by the government, many people have self-medicated themselves by using previous prescription about certain drugs prescribed for various ailments and recently for COVID-19 without consulting the physician or the pharmacist. This is to evaluate the participants practicing self-medication and for what common conditions and common drugs they have been practicing. The awareness is to bring an impact among the individuals, not to follow misconception about certain drugs. Introduction. As we all know that the deadliest virus, COVID-19, has brought a great impact among each and every individual in this whole world. During the pandemic, people remained isolated at their homes, maintaining physical, emotional, and social distancing, practicing hand washing techniques, and wearing face masks to protect themselves. Self-medication is a common practice where people use the medications, whether it be modern or tradition, which they implement it on their day-to-day life. Although the vaccine wasn't developed during the pandemic period, people still practice to self-medicate with or without having the symptoms related to COVID-19 by searching in the internet and media without consulting the physicians. The factors influencing this issue has brought serious adverse effects on many who use old prescriptions and take fake prescriptions to the pharmacy in order to treat their symptoms. Recently, immune-boosting supplements and herbal concoction has become an ongoing trend in treating COVID-19, which is not sufficient enough by killing the virus. This situation has increased regarding misinformation about medication, leading to public confusion and panic and increased use of self-medication. Aim and objective. The main aim of the study is to know the knowledge, attitude, and practice among the people in Chennai practicing self-medication during the lockdown 2020. Objectives. The primary objective is to study and evaluate the participants practicing self-medication during the lockdown 2020 and to find out the most commonly used drugs and most common conditions of using the drugs during this lockdown period. Secondary objective is to bring awareness to the community on practicing self-medication. Methodology. The survey will be carried out by enrolling participants at a locality in Porur, Chennai. Around 323 participants will be participating in the survey through online by sending the forms through their email ID and WhatsApp to record their responses. As it is not feasible to do a community-based survey during the outbreak period, the study will not undergo a door-to-door interaction. It will be a questionnaire-based survey with 22 questions, both in English and local language for their preferences, and their response will be evaluated. The collected data will be analyzed by utilizing the statistical package of Social Sciences version 24. Expected outcome. The study will ensure that it will bring benefit among the individuals taking self-medication during this lockdown period by taking necessary measures to prevent the adverse effects, irrational use of drugs, and not to practice by taking the medications without a doctor's advice. The awareness can bring an impact among the individuals not to follow misconception about certain drugs and administer it, thus causing adverse effects in their health. Thank you.
Good morning, one and all. I am Dr. Rajeshwari, Reader, Biochemistry Department, KSR Institute of Dental Sciences and Research, Tirchangudu. Today I am going to present a piece of research work entitled Evaluation of Lipid Tetride Index and Lipoprotein A in Coronary Artery Disease Patients of South India. This work was done under the eminent guidance of Dr. V. Govindaraju, retired professor and head, Biochemistry Department, Superintendent, Jayadeva Institute of Cardiology, Bangalore. Asian Indians appears to have the highest rate of premature coronary artery disease, although they have low prevalence of conventional risk factors and nearly half of them are lifelong vegetarians. More information is needed on the interaction between blood lipids and coronary artery disease. This coronary artery disease is the major cause of death in developed countries, including India. Recently, lipid tetride index has been proposed by Inas and et al. As the best estimate of the total burden of dyslipidemias. Dyslipidemias includes abnormal levels of serum lipids, lipoproteins, apolipoproteins, and nowadays even concentration is being done on LPA estimations. This lipid tetride index is calculated by the product of cholesterol, triglycerides, and LPA values, and that will be divided by high density lipoprotein cholesterol. Much work is not being done on the association of lipid tetride index and coronary artery disease patients of India. Hence, it is needed the work to be done on the role of lipid tetride index in different parts of India. As I have shown in the slide, the formula for the calculation of lipid tetride index, that is total cholesterol into triglyceride into LPA divided by HDLC. The normal value should be for this should be lesser than 20,000. Coming to lipoprotein A, it was detected by Kareberg. It is a complex assembled from very different components. One is an LDL-like particle that has ApoB100, which is connected to other glycoprotein called as ApoA, which is a water-soluble protein. Many studies suggest that LPA is a powerful and important risk factor for coronary artery disease, particularly when total cholesterol level by HDL cholesterol level is high. It means the ratio of total cholesterol and HDL cholesterol is high. Yet there is a study needed for LPA estimation in coronary artery disease patients of India. So an attempt was made to show the importance of lipoprotein A in coronary artery disease of South Indian patients in our study. This is the diagrammatic representation of lipoprotein A. You can see very well the connection between apolipoprotein A and B100 in LDL-like particle. Coming to materials and methods, the clinical material for this study was obtained from Sri Jayadeva Institute of Cardiology, Bangalore. 203 patients were having coronary artery disease alone were taken for our study. For comparison of these patients, a group of 191 normal healthy age sex matched subjects were included for our study. They serve as control. Lipid profile was carried out by using enzymatic methods, whereas LPA was carried out by using latex agglutination method, and lipid tetrad index was calculated as I have shown the formula. And for biochemical analysis, Hitachi autoanalyzer was used. After obtaining results, appropriate statistical analysis was done. Mean and standard deviation values were calculated. Based on that, students' test was done and then p-value was calculated. If we see in this tabular column, we have higher concentration of LPA and lipid tetrad index values in coronary artery disease patients when compared to healthy subjects, and high density lipoprotein value is low in coronary artery disease patients, and this value is high in control subjects. Coming to conclusion, abnormal lipid profile, high LPA along with high lipid tetrad index values were observed in our study in coronary artery disease patients. This may be due to lower high density lipoprotein cholesterol value and high LPA values found in CAD patients of our study. Investigation of LPA and lipid tetrad index may be considered as markers for the development of coronary artery disease in young patients, and lipid tetrad index can be considered as a screening test in middle-aged people. Thank you.
My title is on awareness, knowledge and attitude about palliative care among general population. Rationally, palliative care is an approach that improves the quality of life of patients and their families facing the problems associated with life-threatening terminal illness through the prevention and relief of suffering by means of early identification and impossible assessment and treatment of pain and other problems. Palliative care is focused on improving the overall wellness of individuals with serious illness. It addresses both the symptoms and the stress of living with a chronic illness. It may also involve support for loved ones or caregivers. Aim and objectives. Aim. The aim of the study is to evaluate the awareness, knowledge, and attitude about palliative care among general population. Objectives. Palliative methodology. Study design. Community-based cross-sectional study using questionnaires. Study duration. The entire study was planned to be carried out for a period of 6 months. Phase 1 selection of study designing a standard data entry format required for the studies, literature review. Phase 2 IEC approval, data distribution. Phase 3 data collection, data analysis, submission of reports, thesis presentation. Study method. This is a cross-sectional study which involves general population. The subjects will be recruited randomly by sending an invitation to participate in the study. The group of random people above the age criteria of 18 plus are selected and asked to fill the questionnaire. Once they accept and fill the questionnaire, the evaluation is done on the basis of knowledge on study. Sample size. Sample size is calculated using a solved online sample size calculator. The sample size is found to be 218 using the software.
presentation on prescription pattern analysis of antibiotic in the community pharmacy. So moving on to the rationally, prescription pattern monitoring studies are a tool for assessing the prescription, dispensing and distribution of medicine. The main aim of prescription pattern analysis study is to facilitate rational use of medicine, irrational use of medicines in a major problem worldwide. The overuse, underuse or misuse of medicines result in wastage of scarce resources and widespread health hazards. Examples of irrational use of medicines include use of too many medicines per patient, which is polypharmacy, inappropriate use of antimicrobials, often in adequate dosage for non-bacterial infections, overuse of injections when oral formulation would be more appropriate, failure of prescription in accordance with clinical guidelines, inappropriate self-medication.
Good afternoon, everyone. I'm here to present my project titled as Knowledge, Attitude and Practice Towards COVID-19 among college students in South India, a cross-sectional study. And my abstract ID number is 84.
Trial version.
Moving on to the rationale. COVID-19 has emerged as a global threat by causing a lot of challenges to the society and healthcare system. Understanding the students' knowledge, their attitude and practice related to COVID-19. People's observance of the prevention measures is essential for controlling the spread of COVID-19, which is affected by their knowledge, attitude and practice towards COVID-19. Moving on to the literature review, Ping Zhu, in his study Knowledge, Attitude and Practice associated with COVID-19 among university students, a cross-sectional survey in China, concludes that most of the Chinese students were informed of basic information and possessed positive attitude and proactive practice towards COVID-19. Jagjit Prasad Singh, in his study assessing the knowledge, attitude and practice of students regarding the COVID-19 pandemic, concluded that awareness and impacts of various information sources on effective outbreak containment, improved community engagement activities and effective communication are needed during widespread disease outbreaks. Ahmed Fauzi, in his study survey data of COVID-19 related knowledge, attitude and practice among Indonesian undergraduate students, concludes that most of the students had a vast knowledge about COVID-19 and its prevention.
Trial version.
Moving on to the aim and objectives of the study. The aim of the study was to understand the knowledge, attitude and practice of college students around South India regarding COVID-19. And the objectives were to assess the knowledge, attitude and practice towards COVID-19 among college students and to assess and provide the qualitative and quantitative information of the study. Moving on to the methodology. The study was a cross-sectional study and the duration of the study was 7 months and 384 participants were included in the study. A 24-item survey questionnaire comprised of demographic characteristics, knowledge, attitude and practice regarding COVID-19 among college students were circulated. For each correct answer, participant received 1 point. A minimum of 6 points was considered to be adequate for each section of knowledge, attitude and practice. And moving on to the patient selection, all consenting participants of previous years and courses were included in the study. Patients above the age of 18 and all gender who are willing to participate by providing the consent to the study were included in the study. People other than college students were excluded from the study.
Trial version.
Moving on to the results and discussion. We are comparing the age-wise distribution. Around 157 participants were between the age group of 18 to 20 and around 168 participants were between the age group of 21 to 23 and around 59 participants were above the age group of 23, above the age of 23. And moving on to the gender distribution, we're comparing the male and female ratio. Male ratio was around 52.86% and around 47% were female in my study. Moving on to the educational qualification of the participants, around 146 participants were from UG degree and around 187 participants were from PG degree and around 51 participants were research scholars. And moving on to the willingness to participants, Around 42 participants had volunteered in COVID-19 programs, and around 342 participants did not volunteer in any kind of COVID-19 programs. While correlating the socio-demographic characteristics and participant adequate knowledge, positive attitude and appropriate practices, an overall of 86% had adequate knowledge, and 85% had positive attitude, and 89% had followed appropriate practices, and there was no significant difference noted among the age groups, but while comparing the gender ratio, there was a significant difference seen in participants following appropriate practices. And while seeing the educational qualification, RCD shows a significant difference in participants having adequate knowledge, and research scholars were found to have more knowledge while comparing the UG and PG students. Moving on to the conclusion, the study found that the majority of college students had adequate knowledge, positive attitude, and appropriate practices has also been followed most of the time. Understanding the knowledge, attitude, and practice towards college students regarding COVID-19. Students with low knowledge about COVID-19 has to be targeted in order to promote their positive attitude and to adopt safe practices. Certain awareness programs for college students to impart more knowledge and also to insist the importance of safe practice during this COVID-19 could help prevent the occurrence or spread of COVID-19 infection. And these are my references. Thank you.
Trial version.
Good morning. I'm going to present a presentation on prescription pattern analysis of antibiotic in the community pharmacy. So moving on to the rationally, prescription pattern monitoring studies are a tool for assessing the prescription, dispensing and distribution of medicine. The main aim of prescription pattern analysis study is to facilitate rational use of medicine, irrational use of medicines in a major problem worldwide. The overuse, underuse or misuse of medicines result in wastage of scarce resources and widespread health hazards. Examples of irrational use of medicines include use of too many medicines per patient, which is polypharmacy, inappropriate use of antimicrobials, often in adequate dosage for non-bacterial infections, overuse of injections when oral formulation would be more appropriate, failure of prescription in accordance with clinical guidelines, inappropriate self-medication, often of prescription-only medicines, non-adherence of dosage regimens. So moving on to the literature review, author Roland Naimak in the year 2019 conducted a study on antibiotic prescription pattern and determinants of utilization in the National Health Insurance Scheme at Tertiary Hospital in Niagara. And concluded that they were under prescription of injections, which is commendable on the other hand. And author Antonio in the year 2012 conducted a study on antibiotic prescription trends in a pediatric population in Lithuania and concluded that inappropriate prescription has been associated with mounting rates of antibiotic resistance. And author Kiranos in the year 2019 conducted a study in understanding physician antibiotic prescription behavior, a systematic review of quantitative studies. In this study, he concluded that the excessive use of antibiotics may result in resistance of bacteria to wide spectrum antibiotic and higher possibility of other infection developing and bacterial becoming more and more resistant. Moving on to the aim and objectives of my study, Aim of my study is to understand the rationality of the antibiotic prescribed in a community pharmacy, and my objectives of the study is to evaluate the rationality of the antibiotic prescribed, drug interaction, adverse drug reactions, dosage intervals in the community pharmacy. So moving on to the methodology, the study is designed as an observational study, and the study duration is like 7 months. So the sample size calculation is done by Ross Soft online sample size calculator for a random sampling of a population of 100 persons with the confidence of interval of 95%. The sample size is found to be 80 with 5.75% of margin of error. Moving on to the next slide, participation selection. In this inclusion criteria, patient with the prescription sheet who were prescribed with antibiotics seeking community pharmacy are selected for the study. Moving on to the result and discussion. This is table 1. In this, the patient demographic is shown. And this is figure 1. In this, number of days prescribed for the antibiotics to the patient in the community pharmacy is shown. And moving on to the figure 2. In this, the number of prescribed antibiotics per prescription for the patients is shown. So moving on to the conclusion, by this prescription pattern of various antibiotic and their rational use will be studied, and the rationality of the antibiotic prescribed, drug interactions, adverse drug reactions, dosage intervals in the community pharmacy. So in conclusion, we believe that these findings will promote a better understanding of physician prescriptions, of the factors that influence the antibiotic prescription process, clarify how such factors influence the decision-making process, and highlight the importance in the design of strategies aimed to tracking the concern effectively. So these are the references. Thank you.
Hi, good morning to everyone present here. I am Jayasudha, research scholar from Biomedical College for Women, Anna Salai, Chennai. My title is Multiple Cancer Protein Targeting with Endophytes and Ethnopharmacological Approach. First, what is Neve molecular docking? It is a method which predicts the preferred orientation of one molecule to second when bound to each other to form stable complex. Docking is the computational determination of binding affinity between molecules, protein structure ligand. This is the diagrammatic representation of molecular docking. Background and objective of my study. An ethnopharmacological approach in drug designing is the next paradigm shift implemented in various complex disease models. Cancer is the most stressful disease because multiple proteins or receptors are playing a key role in rapid cell division, progression, and cellular communication. So, treating the disease is a daunting challenge. However, this kind of complex network mechanism can be addressed by blocking more than one drug target, like aborting cell cycle, blocking mechanism, and receptor channels. Methodology of my work. The current study is focused on multiple drug targets of cancer proteins and enzymes to elevate disease mechanism with the molecular docking compounds isolated from aloe vera and vinca. The computational biology approach of molecular docking was used to target the binding site of cancer-inducing proteins. Introduction. The concept of structure-based drug designing is employed for structurally known compounds and drug targets. In this current study, the drug target involved in complex biochemical network of the pathway such as BRAF, which is reported in RAF, MEK pathway, and also reported in many cancers like melanoma, non-small cell lung cancer, colorectal cancer, and papillary thyroid cancer. Heat shock protein 19, which is involved in cell signaling, proliferation, and survival of cancer cell. And the next is estrogen receptor alpha, which is highly reported in breast cancer. And finally, aurora kinase involved in multiple mitotic events and essentially plays a major role in spindle assembly. These drug targets are docked with naturally isolated compounds from endophytes of aloe vera and vinca to understand the mode and atom level interaction through computational biology tools and software. This is a diagrammatic representation of molecular docking system. First one is database, and the next model-based structural screening, synthesis, experimental evaluation, and ligand receptor structural determination. In second, molecular target, QSAR and QSPR modeling, design synthesis, property prediction, and experimental evaluation, and finally series of molecules have been docked. Materials and method. Drug target retrieval from structural database. The structure of each target are retrieved from protein data bank. In this study, 3D structure of estrogen receptor alpha and BRAF, aurora A kinase, and heat shock protein have been derived from database. Then receptor preparation of dogging is prepared using BioVia Discovery Studio Visualizer 2019. And finally, ligand protein of dogging. 4 compounds are isolated from aloe vera and vinca rosea. From aloe vera, 2 compounds are isolated, stigmasterol and 2-hydroxybutanoic acid, and from vinca rosea, 2 compounds are isolated. One is vincamide and another one is vincapyridine A. Initially, these are sketched using ACD ChemSketch tool, and further analysis with 3D protein data bank format in BioVia Discovery Studio is used for docking. This method is one of the prominent techniques in computational biology and chemistry to screen hits from a database source. The grid-based docking tool, Autodock 4.2 with MGL tool, was employed for docking receptor ligand interaction study. Is generally called as lock-and-key process. The genetic algorithmic search parameters were used for docking in Autodock with a population size of 150 to 25 lakh maximum number of energy evaluation. 27,000 of maximum number of generations with 10 run before docking is a successful complication of autogrid calculation which is needed to be executed. In this current study, 4 drug targets are involved and the grid is placed at different locations such as estrogen receptor alpha box, which is placed in 27.798 in X, 2.653 in Y, and 24.929 in Z with 0.364 Armstrong, and VRAF dimension are 1.299 in X, minus 5.083 in Y, minus 19.1 0.139 in Z with 0.369 Armstrong. Similarly, Aurora A kinase dimension of grid minus 11.551 in X, 16.694 in Y, and minus 14.136 in Z with 0.275 Armstrong, and GSK3 dimension include 16.58 X, minus 62.67 in Y, and 20.35 in Z with 0.375 Armstrong. Finally, among the 10 blends, the best and top rank poses in the highest negative binding energy were reported. Results and discussion. The current drugs are interlinked to each other in multiple cancer-causing pathways. The estrogen receptor is notably reported in breast cancer progression and its co-expression of gene and byproducts associated with other cancer. Suman Chatterjee et al. States that estrogen receptors are one of the HSP90 clans that are validated oncogenic drivers, so the inhibition of role on HSP90 impedes the aberrant behavior in lung cancer cell progression. Comparatively, HSP90 role is more in tumor cells than normal. It is due to misfolding mechanism of proteins that needs more correction than a normal cell. Further, Radha Munkala et al. Reported an intercellular pathway brief about the integrated role and expression of B-type RAP kinase and HSP90 in breast cancer. Previously, the role of Aurora kinase overexpression and gene amplification has known for many cancers such as colorectal cancer, ovarian cancer, breast cancer, and gastrointestinal cancer. This is the protein structure stability analysis. The first one is the drug target, next is the before loop modeling value, and another one is after loop modeling value. Third one is multiple drug target binding of endocytes. The graphical representation of free binding energy on X lab and compounds on Y lab. Drug likeliness property prediction for isolated endophyte compounds. First one is a compound, second one is a log value, third one is molecular weight, fourth one is hydrogen bond acceptor, and fifth one is a hydrogen bond donor. This is a 3D and 2D representation of stigmasterol with BRAF kinase, first A and B, next each of protein 90, which is C and G, estrogen receptor alpha, which is E and F, and Vincombine Aurora A kinase, which is G and H. Finally, results. These compounds were docked to binding cavity of each drug target to identify the potential interaction among the four compounds, stigmasterol, vincomine, and vincapyridine A, binding with arginine residue that communicate the phosphorylation of threonine 288 to the TPX2 binding site, thereby impeding the role of Aurora kinase A mechanism. Similarly, these compounds show an effective mode of binding with BRAF kinase and further targeting with estrogen receptor alpha, and HSP90 is influenced more by stigmasterol than other compounds. Conclusion. Hence, these three compounds shows more prominent towards blocking the various drug targets of the cancer. Besides, compounds that were isolated from a natural source are considered to be safe in treating the disease without side effects. Thus, stigmasterol, vincomine, and vincapyridine A are the potential drug candidate for treating the cancer. Thank you.
Greetings to one and all. Myself Smita Hegde. Today I am here to present my topic entitled In Silico Analysis of Single Nucleotide Polymorphism of Gap Junction Beta 2 Gene in Hearing Impaired Children under the guidance of Dr. Pramod B. Gai, Director, Karnataka Institute for DNA Research, Dharwad, and former Vice Chancellor, Karnataka University, Dharwad, Dr. R.S. Pulugowda, Department of Anatomy, Shri B.M. Patel Medical College, PLDE, deemed to be University. Before going to my research work, let's see the background. Hearing impairment is one of the major sensory disability that affecting millions of people all over the world. Today, 1 in 1,000 newborn children were affected by the hearing impairment, and worldwide studies also revealed that approximately 466 million people, around 5% of the world's populations, were suffering from the hearing loss. Hearing impairment is one of the major burden in a social and the professional life of a child. Connexins have a wide role in the different issues, and thus that mutated form have been linked with the several diseases, including the hearing loss. Connexins are the protein which are included by the connexins are a protein which are encoded by the gap junction beta 2 gene, which are essential for the hearing function. So gap junction beta 2 gene, which is also called a DJB2 gene, which encodes the connexin 26 protein, and this belief that related to the half of the all cases of hereditary deficit. Connexin 26 gene is a member of the connexin family encoding the intracellular channel comprising the gap junction, which allow the direct transfer of the small molecule and ionic current between the cells. With 4 transmembrane domains, connexins are a membrane protein. The hexameric complex of this protein is called connexon. Cell-to-cell channel is formed to form a gap junction between the 2 hexamers in the membrane adjacent cells. Gap junction work as a transmembrane of the small molecules and ions between the cells. The channel constitute can determine which molecule can pass through. Present protein is essential for the maintaining the high K+ion concentration in the endolymph of the inner ear. Sound stimulation of the ossicular chain causes the vibration in the endolymph. K+ion enter the hair cell under the influence of those vibration, and the vibration signals is ultimately converted into the neural signal. This system is regenerated by the release of K+ion from the hair cell to the supporting cell. This is how the hearing function will work. When we come to the GJB2 gene, which is a small gene situated on a chromosome 13, and it has a length of about 5.5 kilobases. There are 2 exons, of which only one contains the exonic sequences, that is 2.2 kilobases long, and translated into the protein with 226 amino acids. In the picture, you can clearly see the gene exon GJB2 gene, which contains the exon 1 and exon 2, and it clearly shows the exon 2 is a bigger compared to the exon 1, and which contains the coding sequence. And most of the mutations of the GJB2 gene are recorded in the exon 2 region. Genomic variation understanding is one of the major challenges of the current genomic research field due to the enormous number of the genetic variation in the human genome. And SNP represents the most abundant genetic variation, ranging between 3 to 5 million throughout the human genome. Mostly the SNPs are in a neutral condition, but some of them contribute to the disease predisposition by modifying the protein function. So our study objective is to find the functional and structural prediction of the 3 missense SNPs, which is recorded in a gap junction beta. So methodology is like that. The 3 missense SNPs I33, B153I, and R127H missense variations were retrieved from our study for the in silico analysis. Pathogenicity prediction of I33T and B153I and R127H missense SNPs done by the proven polyphen 2 PhD SNP and CAD using, and also the deafness variation database. And conservation stated analysis of the mutations was done using the conserve and a prediction of stability done by iMutant software. And structural analysis of these 3 SNPs were done using a Swiss model and Chimera. Pathogenicity prediction for the 3 missense mutations are like that. I33T mutation was found to be pathogenic or disease causing for all the tools which we are selected, which show the deleterious effect, but R127H mutation is showed deleterious only to the few softwares like PhD SNP and SNP Go. Proven polyphen, CAD, or a deafness variation database showed the benign effect to the R127H missense mutations, but the last one, V153I missense variation show the complete neutral or benign effect for all the tools which we are done. Conservation status and the protein stability of these 3 missense variations are like this. I33T mutations show the highly conserved status in the conserve, and it also showed the decreased stability of the protein. In the pathogenicity predictions, also I33 mutation showed the deleterious effect like that. It's in a stability of the protein is also decreased in this protein stability prediction tool, and reliability index is also 8. And V153I mutation is a semi-conserved, and it did not affect the stability of the protein, and the pathogenicity prediction is also show the neutral effect supporting to this. And R127H mutation is it's not highly conserved, but it shows the conserved status, and it is slightly affect the protein stability compared to the I33 mutation. For structural prediction, we only selected I33T and R127H missense SNPs. Because of that deleterious effect, we did not selected B153I missense variation. And according to that, the structural prediction showed there is a variation in the size and charge of the amino acid when compared to the wild type protein structure. So in the picture, you can clearly see the first one is showing the wild type protein, and second two are of that I33T and R127H missense variation protein structure. There you can see the changes in the protein structure, stability, and also there is changes in the charge of the mutant amino acid. By all these, we can clearly tell that through the use of various in silico tools, these are two missense variations I33T and R127H are harmful effect, and the last one V153I showed the neutral effect in the hearing loss. And thank you one and all for the opportunity.
Greetings to one and all present here. I am Saranya Shri, third year BTech Biotechnology student, and I am here to present my project entitled In Silico Analysis Predicting the Effects of a Deleterious Non-Synonymous Single Nucleotide Polymorphism in Human TGF-br2 Gene on its Structure and Function Associated with Pancreatic Ductal Adenocarcinoma. Under the guidance of Srinidhi Ma'am, Assistant Professor, Department of Biotechnology, Rajalakshmi Engineering College, Chennai. Pancreatic ductal adenocarcinoma is a malignancy that continues to present clinicians with significant hurdles in its treatment. When diagnosed, it tends to be advanced, metastatic, and poorly prognosed. It is estimated that it will become the third leading causes of cancer-related deaths in the next decade, and the death toll from PDAC is anticipated to rise further. TGF-beta is recognized as a both tumor suppressor and a tumor promoter, and the current knowledge of the factor that mediate the role of this change and when this shift occurs provides insufficient information. TGF-beta is a vital cytokine in embryogenesis and tissue hemostasis, exerting a large range of functions. TGF-beta expression has been studied in a wide variety of cancer forms, including prostate, breast, lung, colorectal, pancreatic, liver, and skin cancer. TGF-beta levels are positively associated with a favorable prognosis in early stage tumor, while they are positively associated with tumor aggressiveness and poor prognosis in advanced tumors. These observations have shown that TGF-beta pathway has both anti and pro-tumor activity. In early-stage tumors, TGF-beta pathway promotes cell cycle arrest and apoptosis. In contrast, the TGF-beta pathway promotes tumor progression and metastasis at advanced stages. So the figure 1 shows the TGF-beta signaling pathway in PDAC. Any mutation in this pathway may ultimately lead to loss of growth inhibitory effect of TGF-beta and failed repair of genes. In this study, we are mainly concentrating on TGF-br2. A total of 405 NS-SNP were retrieved from NCBI, and all those 405 NS-SNP were subjected to this 6 in silico web tools. And these 6 in silico web tools found 43 most deleterious NS-SNP. This ensured the stringency and precision of the result, and we found those SNPs to be high risk, which all 6 program predicted to be deleterious. Followed by domain identification by Interpro. Interpro was used to identify the position of NS-SNP on the conserved TGF-br2 domain, followed by protein interaction analysis by string database, and pathogenicity identification by Raspody. Raspody is a web tool that is a modern machine learning approach that predicts the impact of human missense variants based on sequence structure and dynamics of protein, and analyzing evolutionary conservancy by Conserve. Conserve tool were used to identify the evolutionary conservation of the amino acids in the protein chain, which performs its purpose by observing the phylogenetic relationship between homologous sequence, followed by analyzing protein stability of deleterious NS-SNP using iMutant and MuPro, and the prediction of post-translation modification sites, and the 3D modeling of NS-SNPs using Swiss model, and then prediction of structural effect of point mutation by Project Hope Server, followed by identification of hydrophobicity of both wild and mutant residues using Discovery Studio. The 43 most deleterious SNPs that were predicted by the 6 in silico web tools were subjected to iMutant and MuPro. These two web tools together found 34 high-risk SNP, which drastically decreases the stability of the protein. Figure 3 shows the major domain of TGF-BR2, where there were two major domains, and one is TGF-BR2 ectodomain, another one is protein kinase domain. Evolutionary conservancy of amino acid in TGF-BR2 protein was generated by conserve. Uh, Using evolutionary conservation and the solvent accessibility, it established the structural and functional residues of the 34 high-risk SNPs of TGF-BR2 protein. All the red and blue highlighted SNPs are highly conserved, exposed, and buried. Remaining are all averagely conserved, exposed, and buried. The three-dimensional structure of wild-type TGF-BR2 protein was generated by using Pyre2 homology modeler, and the protein post-translation modification sites like methylation and phosphorylation was predicted using the respective web tools. The methylation site that was predicted by the software, there were a total of 6 methylation sites, and out of which 5 methylation sites are present in the protein kinase domain, and the one is present outside the protein kinase domain. Mutation in the methylation site may lead to loss of or destruction of gene expression. That may also lead to the non-canonical pathway progression. This may also be a reason for the shift of the protein from tumor suppressor to tumor promoter. Similarly, phosphorylation sites were predicted by GPS 3.0. There are 3 phosphorylation sites which are present in the protein kinase domain. Hydrophobicity of the wild and mutant residue identified by Discovery Studio. And since the hydrophobicity effect is a main driving force in protein folding and binding affinity, changes in hydrophobicity can lead to disruption of normal protein function. The models produced from the Pyre2 homology modeler and the Swiss model was provided to the Discovery Studio as an individual input. The software found all the 34 NS SNP drastically contribute to change in TGF-BR2's protein hydrophobicity. You can see here the wild types hydrophobicity value and the mutant residues hydrophobicity value drastically varies. I'm concluding my presentation by telling that this TGF-BR2 protein has been shown to be involved in pancreatic ductal adenocarcinoma and different tumors. Our investigation may serve as an important guideline in the study of possible diagnostic and therapeutic approaches requiring experimental mutation confirmation and large population-based clinical trials. In this study, the impact of functional mutation on TGF-BR2 protein was assessed using different bioinformatic analysis techniques, which established 32 high-risk mutations with potential functional and structural influence that could be used as a diagnostic marker and could assist in genetic studies with the special considerations of large heterogeneity between different populations of pancreatic ductal adenocarcinoma. Thank you for giving me this opportunity to present my project here.
Hello everyone, myself Rajat Hegde. I am a doctoral student of Laboratory of Vascular Physiology and Medicine, Department of Physiology, Sri B.M. Patil Medical College and Hospital and Research Center, BID deemed to be University, Vijayapura. Here I am here to present a part of my work, the in silico analysis of functional and structural effect of novel missense mutation of NLGN3 gene recorded in Indian autistic populations under the guidance of Professor Kusal K. Das. Okay, let's see. Introduction. When come to the introduction, autism is a complex neurodevelopmental disorder. The autism is mainly characterized by a limited or absence of verbal communications and a lack of social interaction. It may be verbal as well as a non-verbal communications, and lack of social interactions and responsiveness, and a restricted or stereotyped pattern of behaviors. In the first, the term autism was coined by a American psychologist Leo Kenner in 1943. In India, the prevalence of autism is very high. And the recording rate of autism is increasing day by day. But the exact reason behind the high incidence of autism is not yet clear. But more than 1 million cases per year are reported in India. The autism is due to the complex genetic as well as the environmental interactions occurs due to these two combinations of the genetic factor as well as the environmental factors. The many studies have shows that the genetic factors means the genes which are involved in the causing factor or a risk factor of autisms are mainly involved in the functions of brain's development or a normal functioning of the brain. In the previous study, we underwent the genetic sequence analysis of neurologen 3 genes, for example, neurologen 3 gene, neurologen 3, neurologen 4X, and 4Y genes, and we got novel mutations. Here we are presenting only a small data of in silico prediction of novel missense mutations which was recorded in our populations. The objective of this study is to the prediction of the functional effect as well as the structural effect prediction of novel missense mutations. V184A, valine replaces alanine in 184 positions. The neurologen 3 protein. The materialization method, in the previous study, we screened the large populations of 1,828 mentally ill children from all the districts of North Karnataka and identified 136 autistic children. The DNA was isolated from 136 children and we underwent the sequencing of NLGN3 gene using a Sanger sequencing ABI 3500 Sanger sequencing platform. In the present study, we are aiming to analyze the functional as well as the structural effect of these novel missense mutations. The functional effect of this novel missense mutations was analyzed by using a different in silico tools such as the proven PhD, SNP, SNP and Go, and Polyfen, etc. When it comes to the protein stability as well as the conservation status of this mutation residue was analyzed using iMutant. iMutant is a tool which is used to study the effect of this mutation on the protein stability, and the conserve was used to study the conservation status. The structural effect of analyzed by predicting or by developing the mutant protein model by using Swiss model, and these models are visualized and analyzed by using Chimera tool. In the pathogenic predictions of novel missense mutations by the proven PhD, SNP and SNP Go, as well as the Polypen 2, all the five the prediction tools shows that this novel mutations is deleterious or disease causing. You can see in the red color highlight, all the tools show this is the disease causing one. When it comes to the protein stability predictions, these iMutant software shows that this mutation causes the decrease in protein stability. It means it decreases the neurologen 3 protein by affecting through these mutations. And meanwhile, the conservation analysis shows that this mutation means the residue of this mutation is highly conserved. This residue is buried and it is a structural residue. It is having a very important role in evolutionary consequences. It's highly conserved. The mutation in this residue will affect the conservation, will raise the conservation consequences. And in the meanwhile, we developed this mutant protein model by using a Swiss model. The Swiss here we can see in the left side, it is shown in the wild type protein in the green color. In the right side, you can see it's in a mutant protein. The left side protein is a valine is there in 180 fourth position. In the right side, you can see this alanine is there, but there is no difference in the hydrogen bonding between the alanine as well as alanine and the valine. The valine is has a 2 hydrogen bond through 263 phenylalanine. The same 2 hydrogen bonds is there in also with alanine. So there is no difference in the hydrogen bond, but the size and shape of this chart of this wild type and mutant race, the mutant amino acid is different. And these properties of 2 different amino acids will affect the interactions of these proteins. Means the interaction of wild type protein and interaction of mutant protein will vary. The interaction of this mutant protein with other molecules will be deficit, that will be reduced, that may affect the cell-to-cell communications in the new synaptogenesis. So, hence our study is predicted that these novel missense mutations will affect both the structural as well as the functional properties of the neurogen 3 protein. We need to study the in vivo study or in vitro study of this functional analysis. Then only we can definitely say that this mutation will affect the neurogen 3 protein in both structural as well as the functional manner. Thank you.
Hi, this is Vijendra Kulkarni. Yeah, I am a lecturer in the Department of Anatomy at Biomedical Medical College, Bagalkot, and a PhD scholar in the university, that is the Human Genetics Laboratory Department of Anatomy, Sri Biomedical Medical College and Hospital Research Center, BND, that is deemed to be University, Vijayapur. And I am doing my PhD with a collaboration of Karnataka Institute for DNA Research. It is one of the government organizations which is having almost all equipment, including sequencing machines in the Dharwad. I am having MOU or collaboration with this Karnataka Institute for DNA Research for my hemophilia B PhD work. Okay. Today I am going to present a small work from my PhD, that is in silico analysis of novel missense mutation of FIX gene recorded in hemophilia B population of Karnataka. Okay. Main author is myself, Sudhindra Kulkarni, and my colleagues are Rajat Hegde, Smita, Dr. Premendra Kulkarni, Dr. Sanju Kulgi, Dr. Pramod Vijay, Rudrugoda Bulagoda. These all are my mentors. Okay. So before going to start, I want to give an introduction. Okay, coming to the introduction part, hemophilia B is an X-linked bleeding disorder which results due to defects in coagulation factor 9, that is F9. Hemophilia B occurs approximately 1 in 30,000 male births. Using the population of data for 2011 from the census of India, the estimated number of hemophilia patients in India would be around 48,407. Thus, India may have over 70,000 patients with hemophilia A and B. The severity of hemophilia B can be classified as mild, that is 5 to 30%, moderate, that is 1 to 5%, and severe, that is less than 1%. In the present study, we predict the functional and structural effect of novel mutations recorded in the hemophilia B population. My objective in silico prediction of 3 novel missense mutations, that is R43W, E66D, and C157Y of F9 gene in hemophilia B patients of Karnataka. Materials and methods. The retrieval of 3 novel missense mutations R43W, E66D, and C157Y from our previous study. Pathogenicity prediction of novel missense mutations using proven Polyfen2 and SNAP2 and SNAP and Go. These are the pathogenicity prediction softwares. Okay, and the conservation status of mutation residue was analyzed using Conserve tool. That is one of the online tools. And the structural prediction of mutated F9 protein by Swiss model program. So, coming to the part of results and discussion, pathogenicity prediction of novel missense mutations R43W, E66D, and C157Y was analyzed using proven Polyfen2, SNAP2, and SNAP-Go. All the prediction tool shows missense mutations were disease causing or harmful to the function of F9 protein. You can see in the tabular column the mutation that is R43W. For this mutation, proven software is telling it is deleterious. The score is -6.345, and SNAP2 showing very effective, and Polyphen2 is showing probably damaging, and SNAPGO is giving it is highly diseases. So these mutations or the structural changes in the protein, what may it cause in the future? I am going to present this. So like that, E66D has given very horrible results from proven SNAP2, Polyphen2, and SNAPGO. According to this, by seeing the above tabular column, conservation status analysis of R43W, E66D, and C157Y mutation residue showed all the three mutations residue were highly conserved and buried. They have important role in evolutionary consequences. All the three missense mutations causes decrease of F9 protein stability. You can see the reliability index, that is reliability index in it's showing the stability of that particular protein, F9 protein stability. So stability is decreases. Okay. So structural prediction of R43W mutant protein varies in size, charge and shape from wild type protein, and this caused loss of hydrogen bonds and disturbed the folding of F9 protein. E6060 mutant residue was smaller, and this might lead to loss of interactions with the metal ion calcium 4 or magnesium 1 via 4-carboxyglutamate. C157Y mutant residue was bigger, less hydrophobic than the wild type residue. It can cause destabilization of the structure of F9 protein. Thanking you. Any questions? I am coming live. Please interact with me. Please clarify the questions with me. Thank you.
I am Faisal Ali, PhD scholar of biochemistry in Bangalore University. I am going to present my work whose title is Consequences of Polyolosmolites on Structural and Functional Stability of Catalase. I am doing my research at Department of Studies and Research in Biochemistry, Bangalore University. First of all, I'll give the brief introduction about oxidative stress. Oxidative stress is essentially an imbalance between the production of free radicals and the ability of the body to counteract or detoxify their harmful effects through neutralization of species. As a consequence of stress, highly reactive molecules are produced known as free radicals, and cellular proteins are one of the prime targets of these peroxide radicals, as a consequence of which they may proceed to oxidation of side chains, backbone fragmentation, formation of new reactive species, and dimerization or aggregation, unfolding or conformation change, loss of structural and functional activity, and somehow they can change also the gene expression and regulation, which may finally lead to the cellular apoptosis and necrosis. As a consequence of this oxidative stress metabolites on the body, one of the things, if the proteins are the prime targets of these free radicals, the misfolding of these proteins, which are essentially under stress, they may lead to the protein aggregation. This protein aggregation is also the major hallmark of many neurodegenerative diseases, Alzheimer's disease, Parkinson's disease. In Alzheimer's disease, there's the beta amyloid protein, which under stress leads to aggregation of that beta amyloid protein, and there also alpha-synuclein in the Parkinson's disease, and this prion protein in Creutzfeldt-Jakob disease. They may also go under aggregation during this oxidative stress. They may finally lead to various neurodegenerative disorders. So now the question arises that how cellular proteins are stabilized during oxidative stress. Their comes the concept of osmolytes, which are the naturally occurring molecules present inside the cells, which prevent these proteins from misfolding during all these stressful conditions. These osmolytes are actually naturally occurring molecules which are very smaller in weights. They are accumulated in the intracellular environment when cells are subjected to oxidative stress. Osmolytes, and they have been found, they promote towards the folding. They shift the equilibrium of folding towards the natively folded state, and they have been also found to get accumulated relatively high concentration up to several millimolar concentrations in the tissues during various stress. That depends upon the type of the stress. They have been also found they stabilize different types of proteins during various neurodegenerative diseases like Alzheimer's and Parkinson's disease. In fact, polyol class is one of the major class of osmolytes which have so many of hydroxyl groups. They have been found to have some close association with many neurodegenerative disorders like Alzheimer's and neurodegenerative disorders, and they also have been found they have the greater potential for this preventing the misfolding of proteins due to their greater preferential hydration effects. And here there are the 2-3 mechanisms by which these osmolytes they use it to stabilize the protein structures. And one of the in the leftward side there is that mostly denaturants, they mostly shift the transition towards the unfolded state from the folded state. However, these osmolytes they do the opposite and shift the equilibrium from unfolded state to the folded state. And also they do the refolding of the proteins if somehow some denaturant causes the denaturation of the protein. If some osmolyte is dropped to that protein after denaturation, it then leads to the stabilized protein structure. They also have the tendency for the counteraction. That means they can counteract the effect of various salts like guanidinium hydrochloride and urea and some solutes which have the denaturation effect on proteins. They can counteract their effect on the proteins. Our polyol osmolytes of interest that were myoinositol, xylitol, sucrose, and glycerol and sorbitol. Our objectives were first to investigate the effect of osmolytes on the activity, then we performed different biophysical parameters to check for some conformational change in the proteins by these osmolytes, and also we investigated the effect of osmolytes on aggregation of catalase being one of the important protein in the biological system which plays the role in the defense of various free radicals in the body. First of all, we did the activity of osmolytes. We performed enzyme kinetics with the osmolytes, myoinositol, glycerol, sucrose, and sorbitol, and xylitol. We found that except myoinositol, all the osmolytes like glycerol, sucrose, sorbitol, and xylitol, they enhanced the activity of this catalase by decreasing the Km and increasing the Vmax maximum velocity, turnover number, and kcat by Km, which denotes the catalytic efficiency of the protein. However, myoinositol did the reverse of this. It decreased the activity of this protein by increasing the Km and decreasing the Vmax. Also, the turnover number kcat was found to get decreased, and kcat by Km, which is the catalytic efficiency of protein, that was also decreased in a concentration dependent manner. Then we further went to the myoinositol case. We found that there was a close association of this myoinositol with the Alzheimer's disease. In Alzheimer's disease, in some dementia phases, in early dementia phases, there has been found that there's the myoinositol increase in the Alzheimer's patient. However, at the same time, there has been found that catalase expression becomes reduced. So there was the higher gene expression of that catalase protein. However, the active form of this catalase was found inactive. But at the same time, there was increased accumulation of myoinositol. Then we checked whether this myoinositol is responsible for catalase inactivation during oxidative stress of Alzheimer's disease. We found that this catalase in vitro studies, this leads to the structural distortion of catalase. Also, it causes the active site, which is normally corresponded by the heme environment. It also causes the heme environment distortion and the tertiary structure loss of the catalase. And also in vivo experiments, we found that this myoinositol resulted to the enhanced reactive oxygen generation. There was a decreased cell viability and increased cell cycle arrest that finally leads to destabilization of the structure of catalase. So we infer here that myoinositol, due to its aromatic ring structure, induces loss of structure and activity of catalase, contributing towards oxidative stress in pre-symptomatic stages of Alzheimer's disease. Then further we went to another polyols like glycerol, sucrose, sorbitol, and xylitol. We found that and in the study of heme absorption, we found that these glycerol, sucrose, sorbitol, and xylitol, they cause destabilization of the heme environment of catalase. Heme environment is actually buried in the core of the protein inside, which is the active site is also buried inside of this heme environment. It is only because of this oxidation reduction reactions that this catalytic catalysis of this substrate occurs. Further, we went for circular dichorism study in order to investigate whether these osmolytes do have some effect on the secondary structure of catalase. We found that in all the cases, glycerol, sucrose, sorbitol, and xylitol, they causes enhancement in the alpha helical and beta sheet structures of catalase. That means they stabilize the secondary structure by inducing more hydrogen bonds in the catalase. Further, we went for the tertiary structure by using the ANS binding studies using fluorescence. As ANS is actually the dye which binds the hydrophobic patch of the protein. Here we found that glycerol, sucrose, and sorbitol, all the three polyols, they causes compaction of the protein. There was the compaction of the catalase by glycerol, sucrose, and sorbitol in both the intrinsic fluorescence in the leftward panel and rightward panel shows the extrinsic fluorescence ANS binding. In both the cases, we find that there was a Billieu shift of various nanometers with different intensities. In these cases, almost more of the stabilizing effect was found in case of glycerol. Glycerol was having much stabilizing effect than that of sucrose, sorbitol, and this xylitol. Further, we went for the thermodynamic stability. We checked if due to structural stabilization of these polyol osmolytes, do they have some effect on the thermodynamic stability? But we found that this glycerol, sucrose, sorbitol, and xylitol, all the four osmolytes, they enhanced the thermodynamic stability of this catalase to various degrees. In glycerol, the increase of the melting temperature Tm was around 15.17 degrees Celsius, and in sucrose it was 12 degrees centigrade increase, and in sorbitol there was an 11.32 degrees centigrade Celsius this increase of catalase thermodynamic stability, and in xylitol it showed around 7.22 degrees centigrade of this increased thermodynamic stability. Also, we found that in aggregation study by using the light scattering experiments, we found that these osmolytes, glycerol, sucrose, sorbitol, and xylitol, they reduced the formation of aggregation species of catalase. Thus, they have been found like they are best aggregation inhibitors for catalase. So, being catalase as a model protein, we can correlate the same study with other physiological proteins like beta amyloid, alpha synuclein. Thus we can by using these osmolytes, we can prevent the different aggregation formation of different stressful proteins. And the outcome of the study is identification of biologically active molecules that can prevent the structural and functional alteration of antioxidant system disorders that may serve as promising therapeutic intervention for proteopathic neurodegeneration. And also protein aggregation, which is also the hallmark of various neurodegenerative diseases like Alzheimer's, Huntington's, Parkinson's, and other disease. These osmolytes can act as best proteopathic drugs, this aggregation inhibitors for preventing the aggregation of these proteins. Also, we can use these osmolytes for stabilization of improperly folded proteins and intrinsically disordered proteins. And in addition to this, as catalase is having the various biotechnological applications, like it has been used in bleaching processes in industries, we can also increase the shelf life of this protein by using these polyolosmolytes that may increase its stability for industrial use. Thank you so much.
of ERK2 or MAPK1. So here is the crystal structure of MAPK1. Then for further docking analysis, certain bioactive compounds from the Phyllanthus aqueous fruit extract was selected, namely methylprednisolone, tridesacetokivorin, hydroxysalmeteriol, dexatetraenoic acid, and trihomovitamin D3. These were the 5 compounds which were selected based on the biological function. The 2D structures of these bioactive compounds were generated using ACD ChemSketch tool. Open Barbell converter tool is used to convert this file into the PDB format, and that is used during docking analysis. Further, Autodock tools were used to analyze docking simulation. So based on the lowest energy, the best ligand receptor structure was found. So here are the results where the interaction between MAPK1 and trihomovitamin D3 is shown. This figure shows the interaction between MAPK1 and hexadecatetraenoic acid. This third picture shows the interaction between MAPK1 and methylprednisolone. This is with the fourth compound, interaction of the MAPK1 with hydroxysalmeterol. And this is MAPK1 and tridesacetokivorin. This is the overall docking results. From the table, it is clear that the best ligand receptor structure is found to be that of tridesacetoxykivorin, because the number of hydrogen bonds formed is the maximum, namely 5, and the least energy is involved, namely minus 7.79. Thus, it is concluded that bioactive compounds from fruits of Phyllanthus acidus plant may have cardioprotective potential. However, further wet lab studies have to be performed to confirm it. Thank you.
Eastern University, Sri Lanka, for the International Conference, Webinar Conference on Recent Advance in Biomedical Research. I'm here for as a international invited speaker under the title of Scientific Validation of Ayabringraja Karpam Herbometallic Siddha Drug. The main focus of this talk is providing scientific validation of Ayabringraja Karpam, a herbometallic synergistic Siddha formulation, which is commonly used as anti-anemic drug. The traditional medicine from time to time immemorial are found to be derived from natural sources because of remarkable therapeutic effects with less side effects. The classical Siddha medical text mentioned Ayabringraja Karpam for various medicinal properties, especially for the anemia, lethargy, and premature grey. These are the main properties they have mentioned the Siddha text. As per the Siddha quotation and Siddhas has committed, I have selected this drug for this particular drug scientific validation. Ayabringraja Karpam is an important herbometallic synergistic Siddha drug for Pandu, that means anemia, which contain powder of iron or iron, that is called manduram. Then Vedelia chinensis, bringarajam, and citrus lemon, elumichai. So these are the main 4 components to prepare this drug. Actually, it is a rejuvenating elixir, Kaya Kalpam. In Siddhas, they have committed Kaya Kalpam is one of the main mode of preventive therapy. So that means the Kaya Kalpam lengthen the lifespan to be healthy and to avoid premature death. So that if you see that Ibringraja Kalpam, especially I have selected, even though the 3 properties is there, but I have selected only the anemia. Why I have selected anemia and what is anemia? We have to think about that. Anemia occurs when the total number of erythrocytes and amount of hemoglobin is reduced below the normal values of 130 gram per liter in male and female 120 gram per liter, which results from defective cell production, maybe the nutritional anemia, excessive hemolysis, hemolytic anemia, blood clots, the iron deficiency anemia, the role of ABK for anemia with special reference to the iron deficiency anemia, since it is the most common prevalent among the world population. Because that among all the anemia, there are varieties of anemia. I have selected iron deficiency anemia because my drug also contain iron content, and it is the most common prevalent among the world. What is the impact of anemia? It is a silent killer. The patient normally they will not come for the OPD for the complaint, not for the anemia. Even for other diseases, they will say they have diabetes mellitus, they have the hypertension like that. They can say, but they will not say they have anemia. So the patient doesn't know. That's why it is also very silent killer. So when we take the history and the examination, we can found the patient suffering from anemia. It is a gradual onset of public health problem among world population with serious consequences, not only for an individual, but also the socio-economic health. Iron deficiency followed by anemia has high impact on various health implications, which affect the GDP of a country. Because that if the patient is affected, even the silent signs and symptoms, if they go to the work, they couldn't perform it. Then if the worker is not performed, that company or the sector will be affected. And maybe sometime if it is the severe, leading to myocardial infarction, then leading to death. So these affect the skilled people who are working very keen, who are involving in the increase the GDP of the country. With the loss of the people due to this silent killer that affect the country. The WHO report 1993 to 2005 started that more than 1.62 million people were affected by anemia in the world. This condition is inherently poverty, and therefore it is widespread among third world country population. That is mostly affected third world country because of lack entry to proper health care options. It is worsened. That is the main reason. So this is the method of preparations of the Ibringraja Katpam. Iron, then heating process, after that purification process. Then we will get the purified raw materials. Like that, another component of manduram or iron, that is also under the purification process. Then purified materials, and after that mixed together, and using that Vedelia chinensis is used for certain days, and after that lime juice, again Vidalia Chinese is used, and get the waxic consistency. When the waxic, it should be treated with the solar heat, not for any other incineration. It should be the solar heat. When it becomes a waxic consistency, it has to, as per the Siddha quotations, Siddhas as in the post forms, they are given in the quotation, they have mentioned it should be transferred to the herbal cup that made by the Ficus bengalsi dry leaves. Then only after drying on that cup, it is called tonnai. When it fully dried, make it as a powder. I made it as a capsule form. So why I have explained these things, why the Siddhas has committed about to transfer the tonnai, there is a scientific validation behind to that process. So the plan of the today scientific validation of this drug, phytochemical and physicochemical analysis, actually the preliminary screening and determination of the bioactive compounds, physical properties like particle size, zeta potential, BET analysis, and with the sophisticated equipments like XRD, XPS, XRF, atomic absorption AAS, FTIR, same with EDAX, TGDTA. This is the preliminary study I did that. Then I move into that in vitro study, antioxidant activity, membrane stabilization, ferric reducing power, and toxicological studies, because before we give to the patients, we have to find out whether the drug is toxic or not. So in that point, acute toxicity and subacute toxicity in the rats, I have studied. Then pharmacological studies, hematinic activity in the rat, that also studied. Finally, the clinical study with the patients. First, I did the pilot study, then started the proper study. So in that way, I have started my validation, scientific validation to the Ayabringraja Katpam. So Ayam and Ayabringraja Katpam, Ayam and Manduramba heated up to until red hot, as you see in that diagram, and quenched in different type of liquid media immediately until flakes of iron become fine powder, while physical transformation like brittleness, breaking, side reductions may occur. Thus increase the bioavailability and potentiate its efficacy. You know that in our system of medicine, we should do the purification and before go to the process of preparations. In that point, here we are quenching this material, the heating materials, that iron and manduram, to the gingerly oil and nakadi and different sources. Based on that, it get that brittleness, breaking and size reductions, thus increase the bioavailability. Organic compounds of quencher are transformed to the material into organometallic compound, which is favorable to body-friendly materials after quenching, because there are main three processes. What is that? First is quenching, then impregnation or the levication, then incineration. So in this case of Ayabringraja, we are not concerned with incineration, only the solar treatment. Some basic relation to the particular medium, while correlate with organic and inorganic materials. Interaction may be account of absorption, penetration, absorption and formation carbometallic drug. So the first preliminary study find out the bioactive compounds. So the after preparations and we had to do preliminary study and GCMS analysis, FAR analysis also. In that point on the preliminary, you know that screening of ABK as well as preliminary screening of Tonnai raw phycosbenzoin leaves and preliminary screening of Tonnai after processed. After process means that we put this vaccine consistency of the drug to get dried in the solar heat. So what is happening here, if you see that raw materials are the flavonoids and phenol, proteins and sterols, carbohydrate. And in the after process of this drug, this protein is disappeared, phenol also disappeared. Again, it has come to here, the phenol is positive in the final product of the ABK, protein also positive in the final product of the ABK. That means that as per the Siddha's concept, this transferring to the herbal cup, that means the Tonnai, it gives the scientific approach, you know, that protein binding is increased as well as since it is a Kayakalpam, phenolic compound is very important to play as the antioxidant. So it has been transferred. So it is about 18 compounds were identified by the GCMS analysis. Total of 18 structural chemical compounds, various molecular weight. Here that 18 compounds we have got it from the GCMS analysis. You know that two compounds, fatty alcohol and fatty acids, are very high because that ABK shows the presence of 48% iron oxide and 65% organic moieties along with the moisture. So that in that compound, even though metallic compound is there, that is different component, different percentage. The negative zeta potential indicates the ABK sample have bioorganic compounds and its stability and aqueous medium. Because it will come that, you know, that in the zeta potential is the negative, that reduce the reduction of the energy induction because it gives the greater surface potential. So which gives the stability of the drug. So that in that point, the quantitative analysis of the ABK drug shows presence of various minerals along with iron oxide, which are involved in various pharmacological activity in human biological system. In the FTIR analysis, OH stretch, CH2 stretch and CO stretch and OCC stretch confirm the presence of organic moieties in the surface of ABK drug. Here, this is the FTIR analysis graph. This is already I have seen. ECMS analysis. This is the fatty acids and fatty alcohol are very high peak level. So GCMS identified that 18 compound, you know, that terpene and phenol, fatty acids, litapene, plasticizer, marine compound, hydrocarbon, those are identified in the GCMS analysis. Then if you move the physical characters of the ABK, the loss of drying LOD at 105 degrees centigrade is the ideal value. 10 to 20 it should be, but it is at if it is a low, it prevents the damage of the drug and microbial contamination. So in that point, it is very ideal range, loss of drying value. And total ash also that 54.0. You know why? Because of the organic component with the metal and mineral. And it is water soluble ash, this 2.0, bulk density and tap density and flow property. That bulk density is bulkiness, it indicates the bulkiness. And if it is increased bulk density and tap density, reduce the flow property. It is the great effect of the reduce the flow property, then that is a good character of the drug. So that is also good signal to the Ibrahim-Raja-Kappa analysis. And pH is acidic form. So if it is acidic form, we can avoid the contamination of the microbes. And the theta size and theta potential. Theta size is little bit high, that compared to this one, not the high, but it is, they are given the amount, but it is not in the nanoparticles, but in the XRD says it is nanoparticle, because in the theta size evaluation, it is diluted with the in the extract of this water. Then only the organic molecules get absorbed and show the big size. But as per the XRD that and SEM analysis, as per the SEM analysis and EDS analysis, it shows that nanoparticle. But theta potential, as I told you, that minus, that is it is a good signal for the stability of the Ibrahim-Raja-Kappa. In the XPS pattern, short presence of organic matter on the surface of the drug suggests it acts as coating material in the surface of the metallic compound. So that organic matter is available, the surface of the metal, it is well absorbent and adsorbent, so increase the biocompatibility. The macroscopic appearance, same with EDS, showed nanoparticle size of ABK form, association with organic molecules, and enhances the biocompatibility of the drug. XRD pattern of purified iron and manduram, if you see this is the XPS. So there are other components, calcium and iron. XPS pattern, and this is the nanoparticle SEM. It is mentioned in the nanoparticle size. XRD pattern, you know that this is raw iron and raw manduram. This is purified iron and manduram. The final one is the final product. This is after the mixing product. So if we compare the raw iron and raw manduram, that the high peaks are reduced here. Here is reduced. Because you know what happened, that oxides are removed. So that after purification, oxides are removed, it comes as a ferric form. That So XRT pattern of purified iron and Manduram show the low intense peak of iron due to removal of iron oxide after purification process. The XRF study revealed that increased iron content in purified form confirms the removal of oxides from the raw material also. Then AA's atomic absorption XRF also confirms the absence of toxic chemicals. So that before I confirm the toxic material in the animal model, I confirm from this preliminary study, here also heavy metal is free from the heavy metals within the limit. This is the XRF reports. GDTA, that with the temperature, how it has changed. And another point I went that the in vitro study, because membrane stabilization effect of the drug. You know that the free radical and the radical involvement in the thiacalcum. So that because of that, and we have to check about the membrane stabilization effect also. So in this point, RBC membrane stabilization effect of the ABK is high compared to vitamin C. Vitamin C is taken as a comparative drug, and ABK is very high compared to vitamin C. And as well as the ferric reducing power of uses of lemon, Wedelia chinensis, and Eclita alba. Because you know why that in our quotation, in our prose, they have mentioned generally Cadisalai. Cadisalai, there are two varieties, white and yellow. Normally, our Siddha concept say the colored one is the more powerful and the chemical concentration is high. In that point, I try to analyze which is yellow one or white. White is the Eclipta alba and yellow one is Wedelia chinensis. So where that it has the high power that you know that reducing power is significant increased compared to white Eclipta alba. And the antioxidant is not much increased compared with the standard gallic acid. So then I move into the toxicity study. After the in vitro study, I move into the animal study. Toxicity study is done in the you know that subacute and the acute study. Then the subacute, you know, the 28 days. There are 70 animals. Groups are divided. You know that even the normal, and then lime juice. Lime juice is the anubanam vehicle for the Iberingraja catworm. Because of that, we have used that also as a vehicle control. And dose divided into 250, 500, and 1000. We have different doses. Because that in acute toxicity, we have given the single dose 2000 milligram per kilogram. That is the standard. Everything based on the OECD guidelines. We have done that, and ethical with the ethical permission, I have done this. Then I have put the satellite group also for the animal, because to check about the reversal effect. In that point, the if you see that acute toxicity, you know, the 2000 single dose, 2000 milligram per kilogram single dose, no visible signs of toxicity, changes in respiration, circulation, autonomic and central nervous system, behavior pattern were observed during the entire observation period. So it is 2000 milligram per kilogram is a safe dose, is said as per this acute toxicity. But anyhow, I move into the subacute toxicity. In that, ABK confirm fixed safe dose that may not cause toxic effect or threaten the life of the animal. The subacute toxicity study for 28 days with repeated dose at 250 milligram per kilogram 500 milligram per kilogram and 1000 milligram were confirmed the safety of the drug. There was no toxicity sign of mortality observed. And also hematological, biochemical, histopathological changes were not observed in both sexes treated with ibrinrajakatam. In the satellite group, did not show any reversal toxicity in vehicle control as well as the high dose satellite group. So then this NOEL is 1000 milligram per kilogram body weight and considered for ABK is safe for the oral administration. So it is free from the toxic study. It has been cleared through the acute and subacute study. So this is the, as I told this is the hematological parameters in the toxicity study. Everything is normal, nothing disturbed. And biochemical parameters also normal, no any other toxic effect observed. Then histopathological studies, male and female, liver, spleen and kidney were taken after sacrification of the rat. It has been sent for histopathological analysis. Here, that is also, there is no any significant pathological changes during the drug administration. Then I move into the pharmacological study. Before I give the drugs to the patient, I have to check that whether increase of hemoglobin is how far in the animal. So in that point, I have taken these Wilstar albino rats and doses fixed based on the formula 50 milligram, 100 milligram and 200 milligram. It has given for 28 days and evaluated the hematological effect. Here that hematinic activity of the pharmacological study confirmed the ABK has vital iron component and regenerate the red blood cells, hemoglobin and hematological indices showed significant increase in the Wilstar albino rats after 28 days treatment. The observation made regarding dose impact, the 50 milligram, 100 milligram kilogram by body weight were effective for oral administration because we have given that 50, 100 and 200, but there are changes in the 200 milligram because that started to decrease. So effective is only 50 and 100. This is the hematinic activity. We see how that hemoglobin changes has been shown as per the statistical analysis. Then if I move into the clinical study, after finish these all preliminary studies, move into the typical clinical studies. That is my ultimate objective. You know why that ibrinrajakatam is practiced everywhere in Sri Lanka as well as in India without having the scientific evidence. But it is effective. It is found it is very effective and safe also. But if you want to give that message to the scientific community, we have to do this process. Then only that is acceptable. So in that case, 130 patients, 116 iron deficiency anemic patients in both sexes were selected with the 90 to 60 age limit with the typical clinical signs of iron deficiency anemia, IDA, and hemoglobin within the range of 7 to 11 gram. Deciliter were enrolled after obtaining the written consent of the patient. Here also ethical concern taken into consideration. And not only that, we had to register our drug SLCTR in Sri Lanka clinical trial registry. After that only we can move into the clinical trial. That also, this Ibrinraja is registered in SLCTR, then they are given the permission to move into the clinical trial. Out of 116 patients, 102 were participated and completed the study successfully. The patient was asked to attend the OPD once in 10 days to collect the medicines as well as to assess the physical examination, laboratory investigation, performed 3 times, 0 day, the baseline day, the first day, and 48 day, 98 day. Because you know that drug has given only for up to 48 days because the Siddha quotation, Siddha text says only the drug should be given 1 mandala. More than that, not recommended. That is only for the 48 days. So compare with the small doses of modern drug of the iron supplement. This is Siddha's stated 1000 milligram per day. That is 500 mg BD. So in that point, we try to continue the trial up to 90 days without the drug administration. Because to check whether the 2 things, one is there any untoward effects or any increase of hemoglobin because of the releasing from the iron storage, it is occurred or not. For that purpose, without having this drug, continue for up to 90 days. In that point, this we can see that table. What about the signs and symptoms reduced of the patients? The cardinal signs and symptoms were taken to measure: weakness, palpitations, pallor, fatigue, breathlessness, exercise intolerance, and pedal cardinal signs. You know that after the treatment, it has most probably the complete recover, 96, 97 recover. Because that subjective symptoms. And statistically significant improvement of subjective parameters like weakness, palpitation and breathlessness, effort intolerance, and pedal edema. You know that it is proven that ABK, Ibrinraja, are effective in reducing the symptoms of iron deficiency anemia. And if you see, the hemoglobin is the most useful and effective parameter in the objective parameter. So that hemoglobin level in the baseline at 48 day and 90 day after treatment with the ABK found to be effective to increase hemoglobin as well as RBC, PCV, MCH, and MCHC. All these hematological indices. Not only that, serum ferritin level also show significant effect. So if you see that baseline hemoglobin, average hemoglobin for 102 patients at 9.31 and 48 day with the drug 11.11, and without the drug 48 to 90 days, that means the 42 days, 12.03 it has increased. So that in the from the storage, iron is released, that is indicated. And other hematological values also here before treatment, after treatment, it has been given, that is increased. And peripheral blood smear shows improvement here. It is normochromic, normally microcytic, hypochromic blood smear. That means size reduced, color reduced. Then after 90 days, it has become normal. That is shown in this result. So that if we say, therefore, the medical system, the drug preparation involved metallic and mineral processed, double mineral, different stage of processing. The quantitative phytochemical analysis, the presence of various minerals along with iron oxide, which are involved in various pharmacological activity, human biological system, and also nanoparticle size of ABK form association with organic molecules, which enhances the biocompatibility of the drug. In vitro study, ABK formulations of appreciable that RBC membrane stabilization effect, which could be one of the mechanisms for its anti-anemic action. So in the summing up, we can say up to that now that in this light up to that in vitro study, the drug is ideal for the anemia, you know that in the RBC membrane stabilization and other preliminary studies also for the positive. The overall, the patient treated with the ABK has got relief more than 90% for all signs and symptoms reported here, as well as significant increase in hemoglobin, RBC, PCV, hematologic indices. This improvement of peripheral blood smear indicate the potency of ABK in reducing symptoms with hematologic activity. No any treatment-related side effects like nausea, vomiting, diarrhea, gastric distress have been reported during the course of study. You know that in the clinical study, we have to observe any untoward effects. With the criteria, we have to observe. In that point, there is no untoward effect claimed by the patient. And concluded ABK good synergistic therapeutic drug at the dose of 1000 milligram only for 48 days for the management of anemia. So therefore, ABK was validated scientifically through physicochemical characterization, toxicological profile, pharmacological and clinical study to claim as a Ibrance safe and effective drug. Hence, it will open a new avenue for the treatment of anti-deficiency anemia along with preventive care, since it is a natural Siddha formulation as a kayakarpam, which leads to improvement of life also. So that drug is the kayakarpam, it gives the rejuvenation to the body as well as increase the blood capacity. So that will give the support to treat the iron deficiency anemia. Thank you very much for the chance given to me as an invited speaker. So IABMS Conference Organizing Committee and other colleagues who has nominated me. Once again, I thank you all for giving the chance. And I'm also happy to present this scientific validated topic to give a message to the society what the Siddhas has committed in the patients with the evidence. Our evidence is Siddha's quotations. Rather than just a written document, Siddha quotations are strong evidence to us. With that quotation, the scientific validation, it has given the new message to the scientific society. Thank you very much.
Good evening, everybody. My lecture is on honey. Honey is a wonderful gift from nature to mankind. And I'll be concentrating on the topical application of the skin odors, the health benefits which we get. Now, historically speaking, honey was known almost 5,000 years ago, and ancient Egyptian physicians were able to use its medical properties. The ancient Greeks believed that honey could promote virility and longevity. Honey can be traced back to Jin dynasty in the periods of 2000 BC. The nutritional and the medicinal qualities of honey have been documented in Vedic, Greek, Roman, Christian, Islamic, and other texts. Honey has been used in Ayurvedic medicine in India for almost more than 4,000 years ago. So how this honey is produced? It is honey bee, which is the processor of honey of the maker. Honey bee, it is a specific insect that is responsible for making honey. That is Apis mellifera is the species which is responsible for honey making. And this honey is made, for which the basic material required is floral nectar. It is a sweet watery secretion of plants with variable sugar content. Usually it contains sucrose and small amounts of glucose and fructose. Also contains minerals and mostly which is potassium. Water soluble vitamins also present, ascorbic acid along with other B complex members and in trace amounts. Occasionally, plant sap such as honeydew is also gathered by honeybees in making honey. Honey is made in the colonies. Honeybees live in colonies. They are social insects. In that colony, there is one female queen bee around which the beehive is centered, and there are seasonally variable numbers of drone bees which fertilizes the new queen. Almost 20 to 40,000 female bee workers will be there who will be constantly working day and night to make the honey. Now, what is the process of making honey? After collection of the nectar from flowers, it is usually mixed with the saliva by the honeybee, which contains enzymes such as transglycosylase and glucose oxidase. And this mixture is then added to the honeycomb, a small unit in the beehive, and it is constantly evaporated. Water is by constant fanning of the wings by the workers inside the honeycomb until the density of this fluid reaches to about 1.36 kg per liter. That is the 36% denser than water. So the honey becomes thicker and more viscous. The pH of this honey is acidic. It ranges from 3.2 to 4.5, and so the final product of honey is highly viscous with high osmolarity. Now, if you look at the composition of the honey, you can find that honey is a mixture of sugars other than compounds such as sucrose. It mainly fructose. The concentration ranges around 38.5%, and glucose is around 31% of the total carbohydrate present in honey. And the pH of the honey commonly varies between 3.2 to 4.5. And depending on the quality of the nectar and the pollens which is gathered by honeybees, the vitamin contained in the honey is variable. A general composition can be the carbohydrate is around 82.4, dietary fiber is 0.2, protein is very less 0.3, water content is very less 17% only. The vitamins is 0.75, 1 milligrams, and contains B1, B2, B3, almost all vitamins. Minerals is around 68.54, and it includes calcium, iron, zinc, magnesium, phosphorus, sulfur, potassium, sodium, and magnesium. So it is a highly complex mixture, and among the minor components, the honey contains mostly bioflavonoids, and it contains is a variable depending on the source of nectar and also the season. And these bioflavonoids and these phytochemicals imparts a special characteristic color, flavor, and odor to the honey. It also influences the stability of glucose oxidase. So honey may vary as much as 100-fold in potency of its antibacterial activity because of different sources and different levels of hydrogen peroxide, as we can see further. The phytochemicals present in the major phytochemicals present in the honey, in addition to the sugars, it contains traces amount of several phytochemicals such as uh, pinosembrin, uh, pinobanskine, and chrysin, catalase, and inhibin factors. These are some of the phytochemicals which are identified, and it is illustrated in many of the experimental studies. All those phytochemicals mostly act as a antioxidant, and they have antioxidant activity. Now, honey finds wide application in various fields. Mainly, it is a nutritional. Honey is known for its nutritional importance. It's a dietary component. Apart from that, honey also is used as a cosmetic agent. In various cosmetologists have effectively used honey, and the medical importance. The honey is being used in medicine as due to its various properties, such as it acts as an antibacterial agent, as an antibiotic. Also, it has got anti-inflammatory properties, and it is a strong antioxidant components are there, so it is act as an antioxidant. Also, it is an immunostimulatory, therefore, promotion of immune um, powers. Honey is very effectively used. Also, it acts as an antiviral agent. So it has a wide activity, and therefore it is finds application in variety of medicine apart from traditional medicine. Now, the medical use of honey can be further. You can see honey has been used as a medicine since ancient times. As we can see the history, it is used in Ayurveda as a nutraceutical compound, and very recently, the topical use of honey is been proved in various skin disorders such as burn injuries, cuts, bruises, wounds, and diabetic ulcers. Very effectively, honey is used to treat these disorders. Now, honey is, honey is also used in the sore throat and cough, and it is also very found to be effective in various GIT disorders. The limitation of honey is that it seasonally it may cause allergy, and these allergies because of the pollens collected by the bee, which may exert an anti-allergic effect in some individuals, mediated by inhibition of the IgE immunoglobulin binding to the mast cells. So it could be some exceptions also. Not uniformly can say that honey is 100% safe and proof. Now, antimicrobial properties of the honey, which is well established. Honey has powerful antibacterial action on almost 16 species of bacteria. So it is a broad spectrum antibiotic. And the antimicrobial property is mainly due to high osmolarity and acidic pH is around 3.2 to 4.5. And apart from these, there is a production of hydrogen peroxide, which is bactericidal. And there are non-peroxide antibiotic activity is present is due to the methyl glycol and an unidentified synergistic component. This has been detected in many studies. So, in all, can say that honey is very efficient antibiotic with negligible side effects. Now, the honey is being used in treating the skin disorders. It is an ideal wound care agent, and various ways the honey is used for treating the skin disorders. The wound treatment and wound healing is all because of the high viscosity of the honey, which proves as a barrier for microorganisms, and the high osmolarity of the honey, which provides a moist healing environment for the wound healing mechanism. Now, there is a non-adherent water layer is created after application of this honey, and this highly viscous honey draws fluid from the wound and creates an interface of diluted honey, which prevents further spread of microorganisms. Now, apart from this, there is an activation of glucose oxidase when the glucose comes in contact with the water. This glucose is metabolized, and this produces hydrogen peroxides, which will kill the microorganisms. By this metabolic reaction, the pH is decreased because of the production of gluconic acid. This again destroys the microorganisms. This is the second way in which the microorganisms are growth is limited. Now, wound dressings and wound by honey is a very efficient deodorant, and deodorant in the sense that it prevents the malodorous substances. These substances are like ammonia, amines, sulfur compounds. These are formed by microorganisms of the wound from metabolism of amino acid, from the decompressed serum and tissue proteins. So they are limited by honey. There is a rapid removal of these malodorous substances, and there is a more effective and cleaning of the wound. So it is more effective than specialized deodorizing dressings of the traditional or conventional dressing agents. Now this deodorizing is much faster than clearance of microorganism, which cause a malodor. Therefore, honey proves to be a very efficient natural deodorant. Now, if you look at the wound infection, the molecular pathology of wound infection, in an infected wound, which is heavily colonized by microorganisms, there is production of endotoxins, and these endotoxins decreases the growth, repair of the tissue mechanism, and also it will activate the dormant protease, which normally is not activated, but in the course of time in the wound infection, these proteases are activated like collagenase and elastase in the wound tissue, and these activated proteases will digest the matrix of the newly laid connective tissue. Therefore, wound healing is delayed. There is a destroyed cell growth factors, which is vital for repair mechanism. Therefore, in the uncontrolled wound, untreated wound, there is complications are developed. The stimulation of inflammatory response occurs is all because of respiratory burst, which more glucose is metabolized and therefore more H2O2 is produced. These hydrogen peroxides will cause protease activation, and therefore there is a strong inflammatory response is magnified. Now, there is a production of edema also, and this will squeezes the capillaries and they will decrease the blood supply. So it creates more vicious environment and circle, and wound management becomes necessary. Now, how the honey is works on the wound treatment. Now, the proteolytic activity of the various components of the honey will digest the fibrin that holds the pus on the wound and that creates the wound cleaning mechanism. It is the autolytic debridgement mechanism which honey works. Now, honey also will cause plasminogen activation and it will produce more active plasmin, and this again in turns start fibrin lysis and fibrin digestion. So the debridging action of the wound is a valuable feature which removes the material harboring the large pool of microorganisms and cleans the wound. The anti-inflammatory action of the wound is also an important feature. There is a direct anti-inflammatory bioactivity of the honey because of the various antioxidant phytochemicals present in the honey. This anti-inflammatory activity response is magnified by antioxidants. Normally, this anti-inflammatory response is because of the creation of oxygen-free radical from phagocytes. The topical application of the honey, which contains the antioxidant, will decrease this inflammation by decreasing the oxygen-free radicals. So the activity of free radicals are decreased naturally. These inflammatory complications are decreased. In the past, various clinical trials of honey dressings on burn wounds have been done, and it is papers are available. Research work is carried out already, and they have compared the partial thickness burn wounds versus full thickness burn wounds, and they have found remarkable results. They found that there is a decreased scarring in these honey dressings, honey treated wounds. Is all because of overproduction of collagen by overactive fibroblasts and also proliferation of the fibroblast stimulated by oxygen free radicals. This has been prevented. That's why there is a normal generation of connective tissue and then wound healing is normal. We have worked on this topical application honey, and our work was published in Annals of Burns and Fire Disaster Journal. It is published by Mediterranean Burns Council in 2001 and in 2003. And these papers have received several hundred citations on which the people have worked. Apart from this, there are several publications serially then were published on the literature for which they have received several citations. Now, anti-inflammatory actions of honey is again can be highlighted because of stimulation of leukocytes, which releases the cytokines. The stimulation of immune response to infection is because of proliferation of B and T lymphocytes and its action on phagocytes also. So the honey decreases the WBC in experimental wounds in various experimental models. It has been well documented. The edema and exudates are decreased by honey, and the wound healing is much faster. And honey is responsible to stimulate the angiogenesis by various growth factors such as TNF alpha, and also these growth factors will proliferate the epithelial cells and the fibroblasts in the wound. Therefore, there is a faster healing in the wounds which are treated by burns. In the several experimental models, infection-free wound in mice, it has been documented that honey versus controlled moist saline dressings, it is found that those dressings with honey is results into 114% greater extent of epithelialization and almost 69% greater thickness and granulation occurs when you use honey dressings as compared to the controlled moist saline dressings. So this is a very good evidences which have been available from the literature that honey is very efficient and very faster healing agent. Stimulation of angiogenesis occurs because of the growth of new capillaries in the wound, and therefore more blood supply is there, more growth factors are available, and therefore faster epithelialization occurs. The acidic pH that is around 3.2 to 4.5 of the honey is responsible for increased hemoglobin deoxygenation. Therefore, more oxygen is available, and therefore more and more metabolic processes are faster. So the glucose present in the honey also acts as an energy source for the epithelial cells and for the phagocytosis. These factors add on to the healing process. Now, treating the wound by honey is a painless wound management. When I say painless management, that honey decreases the factors released as inflammatory response, sensitizing the nerve endings. So that's why the patients feel less pain. There's a soothing action. If used in the first, it is used as a first aid for burns and scalds. If you apply honey to this wound, then the pain is relieved in almost 20 minutes. The patient will not find it irritating or painful. The bandages applied around the wounds. The blistering and the ongoing painfulness do not occur if you use honey bandages. Also, it prevents tearing of a new growing tissue when you remove the bandages at the time of redressing. What happens in the conventional bandages using a silver sulfadiazine ointments, there is a tearing of the new tissues, and therefore again healing process has to restart. But this does not happen in case of honey dressing. So using honey dressing is almost painless, very smooth, well tolerated by the patients. Now, I've been highlighting the all benefits and positive aspects of the honey, but again there are certain limitations in using this. Do not say that 100% safe, and because honey, there is a variability in the therapeutic qualities. There is variability in the antibiotic activity. It is varies range from low to high. The phytochemical is all because of the variable phytochemicals content, and also the endogenous catalase level from the various sources of honeys. So it depends on the various sources, what sources are there. Sometimes it is very beneficial, sometimes it is the activity is low, therefore it may not be effectively works. So the antibacterial activity of honey is very broad spectrum. There may be some bacteria which will not be controlled. Another limitation is there may be contamination by some pathogenic bacteria like Clostridium botulinum spores, which is present in soil, air, dust, agri products. And also the sterilization, these all can be removed by sterilization. Therefore, this contamination may not be a big problem. But the other limitation is allergic reactions. Since honey bees collect nectar from flowers, they also carry pollen grains from these flowers, and these flowers may be from various species of plants, and the honey they are produced in the honeycomb. It contains these trace amounts of these pollen grains, and these pollen grains may exert anti-allergic effect in certain individuals. All it is mediated by inhibition of immunoglobulin, which is binding to mast cells. So there is always a risk involved in experiencing the anaphylaxis, as an immune system reaction may overweigh any potential allergic relief. So we have to keep it in mind that before treating, this aspect has to be considered. And other limitation is you may not get pure form of honey unless you're sure that it is collected properly and it is tested and that it is sterile. Then only clinically it is to be used. Then adulteration is a great problem. Commercially available honey may not be pure. Reliability of the purity you must ascertain. And apart from that, because of all this limitation, the honey is yet to get approval by FDI as a standard drug or a therapeutic agent. Apart from the limitations of honey, the benefits derived from honey, health benefits, is much more than its any potential risk. Therefore, there is a great opportunity for future research work. There is a need to elucidate the detailed mechanism of the bioactivities of the important honey component, that is phytochemicals, how they actually work and show its therapeutic effects. Also, the practical approach and more novel methods and wound dressing then should be developed. This will benefit to the patients in general. And also the cost benefit analysis has to be carried out so as to decide and apply the most economical and most effective treatment modalities in the wound management. And overall, the practical approach in the basic research in honey is should not be overlooked as is the basic in the translation medicine. I am very thankful to the organizers of this 41st annual conference, in which I am able to speak. Especially thanks to Dr. E. Padmini, the organizing secretary, who has included my talk in this conference, and also to Dr. G.K. Paul, the president of IABMS. I also wish to thank all the office bearers of IABMS. It's an opportunity to meet, even though it's a virtual conference, but feel like sharing many of research work. I'll be very happy if someone picks up some aspect of honey for his future career. I'll be very happy if someone picks up and then gets inspiration from my talk. Thank you very much again. Thank you.
More than 3,000 years ago, early societies recognized that the diversity of plant life around them could be used for the treatment of human illness. Humankind has explored and exploited every possible resource on the land. In the future, similar explorations of the world oceans using sophisticated equipment, modern chemical and molecular and genomic technologies will uncover a rich treasure chest of new medicinal products, cosmetics, foods, industrial chemicals, and new environment-friendly industrial processes. As you all know, now about 45% of the best-selling drugs and 37% of the drugs in the clinic today are derived from natural products. Thus, natural products have played a crucial role in the drug discovery effects over the past 4 to 5 decades. In that context, there have been concerted efforts to utilize various food sources of marine origin to cater the demand from the consumers who are eagerly looking forward to have better nutrition through dietary interventions. In that context, ocean provides diversity of photosynthetic micro and macro algae, which have been surviving in the planet in spite of all drastic climatic conditions. So they are robust and they are rich resources for the mankind. And among the natural products, nutraceuticals and cosmeceuticals are in high demand world over. Nutraceutical, which is commonly referred to as functional foods, are substances that can be considered as a food or part of food which provides beneficial or health benefits of medical importance. If you calculate the global nutraceutical market, it's quite huge, to the tune of around US $247 billion in 2019, of which India has a strong share of around 2%, which is to the tune of 2.7 billion. In the case of cosmeceuticals, it's also comparatively in high demand, mainly comprising of cleansing, beautifying, and also promoting attractiveness to mankind, and also altering the appearance without affecting the body structure or functions. In fact, the cosmeceutical market in the world during 2018 is estimated as $37.41 billion US. Marine environment, especially coastal environment, provides quite a diversity of macroalgae. So both red, green and brown algae, which provides diverse category of natural products like agar, algin, carrageen and ficardin, laminar and vulvan, and also wide range of lectins and also unsaturated fatty acids and antioxidants and fine chemicals and vitamins. In fact, in India, there are around 841 species of macroalgae are currently reported. Very high diverse algal world is available around Indian coast. In fact, this is a glimpse of a couple of dominant macroalgae present in the ocean, like sea lettuce, Alva lactuca, like Hypnea muciformis, Turbinaria ornata, which are brown algae, and also another brown algae called Commonia savaniosa, which are all few representatives of Indian marine algae. In addition to that, Western world has Fucus, Laminaria and Chondroscapus, widely used in food as well as nutraceutical and cosmeceuticals. Let's see some of the examples of the cosmeceuticals as well as nutraceuticals derived from the marine algae. First, antioxidant. So it is in high demand in the among the human population. So as you know, oxidative stress is a primary cause for the development of various human chronic ailments, including cardiovascular diseases, cancers, and also neurodegenerative diseases. Among the macroalgae, brown seaweeds are abundant among the southeast as well as Indian coast. In fact, most of the brown algae has high concentration of algal polyphenols. So, which is if it is a terrestrial plant, they are derived from gallic and ellagic acid. In the case of the phenols of algal origin, it is derived from the polymerized chloroglucinol. So these are primarily synthesized by marine algae or seaweeds for antioxidant activity, mainly to protect them from UV irradiation. Commonly, they have rich fluorotannins. This shows about the antioxidant activity of diverse fluorotannins like chloroglycinol, ecol, and diacol, and catechin diverge, and the radical scavenging activity, hydroxyl radical scavenging activity, superoxide dismutase. And as among them, fluoroglycinol ranks the first with very high radical scavenging activity. So in future, the seaweeds can provide a panacea for the antioxidant nutritive additive. So allergy. So almost 1/3 of the general population in the world is every day affected by the allergic reaction, mainly because of the environmental problems as well as food habitats. So it is dramatically increasing among the human population, leading to diseases like asthma and also atopic dermatitis and rhinitis. These are all within the last 2 to 3 decades increasing and leading to the great morbidity among the human population and also leading to mortality. And in fact, hyaluronidase enzyme is reported to play a major role in allergic reactions. And brown algae, sargassum, wide variety of sargassum, especially sargassum tenerimum, provides fluorotannin, and these extracts of fluorotannin derived from the sargassum inhibits the hyaluronidase, and IC50 is reported to have around 25 milligram per ml. So as you can understand that it's more potent than the commercially used anti-allergic drug disodium gromoglycate, so which is having around IC50 39 milligram per ml. So which is quite similar to the natural inhibitor catechin. And similar to this, there are wide array of ecol, diacol, and also fuco-difluoroethol. All of them are having anti-allergic activity. These also has the capability to inhibit histamine by modulating the binding between immunoglobulin E and FRI receptors. So micro algae like blue-green algae, Porphyrdinium, Dunaliella, they also have anti-allergic activity, which has the capability to inhibit hyaluronidase with IC50 of 190 and 150 milligram per ml. Further to this, osteoporosis. So osteoporosis is a skeletal condition characterized by the decreased bone mineral density, so which can ultimately lead to frequent or the risk of bone fracture. So among the marine algae, there are several products currently available in the market, especially the marine algae called cystophyllum, which is having capability to absorb algal calcium with the effect of improving BMD in elderly people. So the absorbable algal calcium is a mixture of active absorbable calcium, which is prepared by using the algal ingredients, heating with algal ingredients along with the oyster shell. So such a way that administering the extract could supplement the calcium. And also fucoxanthin also plays a very major role in the treatment which is induced by apoptosis accompanied by activation of caspase 3. So in the osteoclast like cells. So there are several in vitro studies proved that the fucoxanthin suppresses the osteoclastogenesis in human population. Currently, there are also several panacea for anti-inflammatory activity. So the anti-inflammatory, inflammation is the major pathophysiological condition associated with several disorders like arthritis, neurodegenerative diseases, cancer, diabetes, and also cardiovascular disease. So few macroalgae, especially green algae and brown algae, are having natural products, metabolites, which can significantly reduce the inflammation. So here diacol is used for the treatment of the arthritis, and also diacol is a blue-green, fluorotannin, and fluoroglucinol also exert anti-inflammatory activity. In fact, currently there are few products developed by the Central Marine Fisheries Institute. One of them is green algal extract, which is derived from Alva lactuca, which is currently available in the market, which is used for treatment of the arthritis in aged patients. Anti-diabetic, diabetic mellitus is a prevalent common metabolic disorder in the human population in developing and developed world. So in world, it is one of the most serious health concern, and controlling the blood sugar level is very important in the hyperglycemic patients. And in fact, the regulation of alpha glucosidase inhibitors are a cost-effective means to prevent the progression of diabetes. Here also the brown algal polyphenol, fucodifluoroethyl, and also diacol and fluorofucofuroecol. So these are all having high biological activity, which inhibits alpha glucosidase enzyme. So the IC50 varies between around 10 millimole to roughly about 49 millimole IC50. So they are currently effectively used in regulating the anti-diabetic activity. Obesity, so almost around a billion population are considered as overweight adults, of whom more than 300 million are obese, mainly because the economic growth and modernization, urbanization and globalization of food markets are the major contributing elements towards the obesity epidemic. In particular, obesity causes an imbalance in the level of adipocytokinase secreted by the adipocytes. So, especially leptin, adiponectin, resistin, and plasminogen activator enzyme 1. Due to the excessive accumulation of the visceral fat, which can cause the obese syndrome or the metabolic syndrome connected with the obesity. So here also the fucoxanthin, which is the carotenoid derived from the common marine algae Undaria pinnatifida, which is commonly used as the treatment for the anti-obesity. Fucoxanthin has an anti-obesity effect by modifying the UCP1, so expression in white adipose tissues. So in mice, they have tested very positively, and are currently it's in human clinical trial. So in fact, the fucoxanthin rich seaweed extract also used on body weight gain as well as lipid metabolism. In addition, currently sodium alginate derived from the brown algae, mainly laminaria, is marketed worldwide for the weight loss supplement, but its effect on gastric motor functions and satiation is also looked into, or the effect is under investigation. Photoprotective effect. So sunscreen compounds otherwise are prevalent in many of the marine organisms, especially those algae that is exposed in the tidal environment. And number of photoprotective compounds such as cytonemins that is present in blue-green algae, and also microsporins present in fungi, as well as microsporin-like amino acid MMAs are present in micro as well as macro algae, and also few corals and other sponges and other organisms in the environment is having the MMAs, and also phenyl propanoids as well as flavonoids. Flavonoids are the major photoprotective agent in higher plants, and melanins from human. So these are all very important in regulating the UV-induced or UV radiation-induced damage in the organisms. So there are several investigations around the globe to find effective photoprotecting agents. So in fact, microsporin-like amino acids are prevalent in red algae and green algae. Here, Porphyra umbilicalis and porphyridium cruendum are having huge concentration of MAs, which can give protection or protection from UV radiation. Neuroprotective effect. So it's a mechanism and strategy used in order to protect neural cells against injury, apoptosis, dysfunction, and degeneration of the CNS, which leads to the limiting neuronal dysfunction or death after CNS injury. In fact, there are several marine algae derived natural products which is acting as neuroprotective agents. So especially here fucoxanthin, so the pigment derived from Hypnea fusiformis inhibited the expression of NME and cell cyclic progression of GOT0 cells, human neuroblastoma cell line. Pheophytin, alpha-fluophyophytin and its analog, vitamin B12 derived from Sargassum fulvellum are reported to have promoting the neurite growth in pheochromocytoma cells. So such a way you have neuroprotective agents, pigments, and many pigments are currently being investigated for human health. So dietary fiber. So every human population currently needs supplement of dietary fiber, which help the reduction in the incidence of colorectal cancers and also suppression of bowel inflammations and related abdominal disorders, and also easy movement of bowel. The dietary fiber is quite helpful. So in addition to that, it encourages the action of probiotics and growth promotion of health promoting gut microflora. So if you take about the marine algae, the dietary fiber content of seaweed varies from 36 to 60% based upon the dry matter. Nearly 50 to 70% of the total dietary fiber is represented by the soluble fiber fraction. And if you look into the list of the algae, quite a large diversity of algae has soluble fibers, insoluble fibers in them. And some of them have, say, Icenia bichlis, which is very commonly present in the Japan. It's having almost around 60% of the soluble fibers. The sea lettuce commonly present in our coast, and also the nori enteromorpha present in our coast. They also have a significant amount of soluble as well as insoluble fibers that's available. And many brown algae and red algae also have the dietary fiber, soluble and insoluble fibers. In addition, the variety of soluble fibers, which is form of hydrocolloids or agar, carrageenan, algin, fucoid and laminar and porphyrin and olivin, or the diversity of fibers, soluble fibers available from the red, green and brown algae. And if you make a comparison, the papaya has around 5.2 grams of the fiber when compared to Alva leuctica, which has almost around 5.3 gram, which is higher than the content that is prevalent in the papaya. And wrinkling. So with the age, the wrinkles settle. Mainly the matrix metallinoprotease enzymes are very important in digestion of extracellular matrix components and leading to the wrinkle formation. Several types of matrix metalloproteinase enzymes such as MMP2 and MMP9 is already reported from humankind. So inhibition of the MMP is available from marine-derived natural products like phyto-oligosaccharides, flavonoids, polyphenols, and fatty acids. They are all previously reported to have anti-wrinkling activity. In fact, there are few anti-wrinkling creams are also in the market, mainly the pro-collagen marine cream, which is derived from the brown algae Padina pavonica. So Elemis is marketing this particular one. Antiacin, so in the younger population, the acin is a very common ailment, and Propionibacterium is the major causative agents which inflict the pimples in human face. So, in fact, Sarcofuran from marine brown algae, Sargassum macrocarpum, is shown to have very potent anti-acin activity against the Propionibacterium acinus. So, in fact, this treats the sebaceous follicle, act on the sebaceous follicles and regulate the infectivity of the bacterium. So, umpty number of hair care products are also available. In fact, human body surface involves approximately 5 million human hairs in the adult, and the scalp around contains 100,000 to 110,000 hairs at about 175 to 300 hairs per centimeter square. So, there are in aging population, hair loss is a common problem. So, not only hair growth treatment, but also hair products related to the protection of the hair loss, hair care, hair management are currently in the forefront worldwide in the cosmeceutical products. So here a few marine algae like Palmaria palmata, Ecklonia, Undaria, so Eukemia, all very common in the Japanese region, as well as Irish moss, Chondrus crispus in the European continent, and Porphyra and Sargassum from Indian subcontinent. They all have natural products which can protect hair. So, in fact, there are a B group of vitamins such as biotin and vitamin B12 are known to strengthen the hair, and which is rich in the brown algae. And in addition to that, currently there are many patents available in the pharmaceutical, cosmeceutical market, mostly extracts derived from the Palmaria, Ecklonia, Undaria, Chondrus crispus, Laminaria, Porphyra, Sargassum. All these contains active ingredients which can protect hair loss as well as promote hair growth. So, ultimately, the marine algae are considered to be panacea for for deriving cosmeceuticals as well as nutraceuticals, which can be great renewable roses.
Starting from USA, then Saudi Arabia, Kyrgyzstan, Sri Lanka, Malaysia, and all parts of India. It was indeed a unique experience to each one of us. Moreover, this COVID-19 has taught us how to communicate effectively rather than commute to attend these conferences. With these few words, I heartily welcome each one of you to this valedictory function. I welcome Dr. Sri Kumar Chakravarti, Director, Research and Innovation, Masa University, Malaysia, to preside over this function. I welcome Dr. E. Padmini, General Secretary, IAPMS, and also the Organizing Secretary of the conference, to announce the prizes to the winners of oral presentations and e-posters. Biomedicine is one of the prize sponsors. I also welcome Dr. Vetriselvan Subramanian to this program. Thank you. Jai Hind. Vanakkam.
Good morning. My name is Sri Kumar, and I'm here to discuss a few basic fundamental concepts in cancer and the role of genes in cancer. This is not only a revisiting of basic fundamental concepts, but also a way in which we can see how various types of genes can affect the possible outcome of a cancer in any way. Now, I'm going to start with a small quote and a poster from a movie that came about 10 years ago. This was from an X-Men movie called The Apocalypse. And I'm sure you remember this poster which says, "Only the strong will survive." Now, as much as this movie is all about X-Men and mutants and mutations in people, what I'm going to talk today is a little bit about mutations within ourselves. So I'm going to relate a lot of what I'm going to talk today to this movie. This movie is quite a fun fiction movie, but it's quite enjoyable. I'm not encouraging you to see this movie, but probably I'll link a lot of genes with this movie at this point in time. So let's move on. Now, when you see this slide, it says only the strong will survive. So that is how it happens in the human body as well. It's only those with a strong genetic system and a strong immune system who can survive. And let's see how it goes. Now, everything about the strong immune system is related to the gene. Now, whatever is there in the gene helps to control our body. In this case, any mutation at any point in the gene affects the body. And that is what we mean by the strong will survive. If the gene is strong, we survive longer. I don't say that we're all X-Men and we would become X-Men later because of numerous what I mean is how genes can cause a cancer. Now, we all have genes in the body. I'm going to actually go through some basic fundamental concepts where we decide how genes can affect the cancer causing mechanisms in the body. Now, before that, let me actually take you through how a gene sits in our body. It is actually nestled very perfectly. Now, here you see a cell and a nucleus. When you zoom into the nucleus, you see the chromosomes. As you all know, we have chromosomes which form an important constituent in the nucleus. Now let us slice the chromosome. Once you slice the chromosome, you will see very tiny particles which are the DNAs. Now DNA is the main backbone of what we are. And when you break up the DNA, you will see the nucleosomes, and genes are just very tiny components in the DNA. And when you see this picture, you will realize that genes have a nucleotide base pairs. Now, I've broken down the nucleus into 6 levels, so you can imagine how small the DNA or the nucleotide base pair is. It is even tinier than the tiniest of bacteria.
Evening to one and all.
Since it is so tiny, we need to realize that the DNA between individuals is
Evening
identical
to
in
one
most
and all.
cases. Now, for all of you who are listening to this talk of mine, I can guarantee that your DNA is identical, as is mine. But all differences are less than 0.1%. So, now if you take an example of Iron Man,
This part of
Robert
the
Downey Jr., and
conference is
Sylvester
very
Stallone,
interesting
both
and a
are
very
totally
happy
different
moment because the
superheroes,
prize winners
actors in
are
Hollywood,
eagerly
but
waiting, and it is going
almost
to
all
be
the
a
DNA is the
very
same.
interesting
There is
session.
just one small
First of all,
variant
my
between
wholehearted welcome
C
to
and
all the
G or A
participants
and
of
T,
the
which is the
41st
difference
Annual
between them,
Conference
which
of
makes them different
IABMS.
people. And that is the difference between all the people among the 6 billion people in our planet. I know it's hard to believe sometimes, but that is the truth, which means all of us are almost the same. But the reason we are different is because of a very, very, very tiny break in the DNA. So, what are we looking for? Now, I'm sure all of you have smartphones and all of you have Google Maps.
It is a very great, delightful evening to announce the prize winners. Indeed, it gave me an immense pleasure to host this evening. Evening to one and all. This part of the conference is very interesting and a very happy moment because the prize winners are eagerly waiting, and it is going to be a very interesting session. First of all, my wholehearted welcome to all the participants of the 41st Annual Conference of IABMS. It is a very great, delightful evening to announce the prize winners. Indeed, it gave me an immense pleasure to host this online conference despite so many hurdles during the preparation. With divine grace and good technical support, we were able to conduct the conference of this caliber, which is first of its kind in so many aspects. Because of the design, it was 100% seamless, which was our prime aim. This conference truly showcases the commitment that all of you had shown towards the scientific deliberations that were presented for the past 2 days. After the COVID-19 pandemic, the students responded to our call in large numbers. There was participation in all the 4 themes: herbal, bioinformatics, advanced biochemistry, and artificial intelligence. I am to announce the winners of the papers and poster awards under each of these categories mentioned above. The winners have set a very admirable task for other participants for the participants for other participants for the conference. We hope and wish you will continue to improve in your brilliance in your respective scientific career and in other spheres. Now, Dr. other participants for the conference. We hope and wish you will continue to improve in your brilliance in your respective scientific career and in other spheres. Now, Dr. Paul, Dr. Tirunala Sundari, Dr. Rajgopal, Dr. Venkatraman, and Dr. Subhatra Ghosh, Dr. Vetri Selvan, and Dr. Vijay Kumar Kutala assess the presentations of all the participants with the following criteria. The criteria was for originality 20 marks, organization of the content 20 marks, clarity of the work 20 marks, knowledge of material 10 marks, neatness and overall presentation 10 marks, questions and answers 15 marks. So with all that, finally the results have come. Before I announce, I want to thank for this opportunity and also thank my EC members for their support and encouragement. I would like to acknowledge my research scholars Ragni and Radhika, the web manager and Dinesh from Cisco, and my friends and family members for their support. With all this together, cumulatively made the online conference app happen at such a grand level. So the prize winners, as we announced earlier in the circular, the awards will be presented by Biomedicine and Cosbeer. For the Biomedicine prize, winners can publish a paper to be submitted within 3 months, and publication fee of rupees 6,000 will be waived by Biomedicine. The first category is Team 1, that is Bioinformatics and System Biology. The first prize goes to Pita, PG Vaishnava College, Arambakam. Second prize goes to Ajit Kumar, Bharatiyar University, Coimbatore. We go to the second theme. Second theme is Artificial Intelligence and Healthcare. First prize goes to Indranil Manna, Napoya Deemed University. Second prize goes to Mukilin, Wales Institute of Science, Chennai. Congratulations to the winners. Sub-theme 3, Herbal Medicine and Drug Discovery. This audience who are given now, this is for your present, for you to view and appreciate. evening to one and all. This part of the conference is very interesting and a very happy moment because the prize winners are eagerly waiting and it is going to be a very interesting session. evening to one and all. This part of the conference is a very happy moment because the prize winners are eagerly waiting, and it is going to be a very interesting session. First of all, my wholehearted welcome to all the participants of the 41st Annual Conference of IABMS. It is a very great, delightful evening to announce the prize winners. Indeed, it gave me an immense pleasure to host this online conference despite so many hurdles during the preparation. With the divine grace and good technical support, we were able to conduct the conference of this caliber, which is first of its kind in so many aspects. Because of the design, it was 100% seamless, which was our prime aim. This conference truly showcases the commitment that all of you had shown towards the scientific deliberations that were presented for the past 2 days. After the COVID-19 pandemic, the students responded to our call in large numbers. There was participation in all the 4 themes: herbal, bioinformatics, advanced biochemistry, and artificial intelligence. I am to announce the winners of the papers and poster awards under each of these categories mentioned above. The winners have set a very admirable task for other participants for the conference. We hope and wish you will continue to improve in your brilliance in your respective scientific career and in other spheres. Now, Dr. Paul, Dr. Tirunala Sundari, Dr. Rajgopal, Dr. Venkataraman, and Dr. Subhadra Ghosh, Dr. Vetri Selvan, and Dr. Vijay Kumar Kutala assess the presentations of all the participants with the following criteria. The criteria was for originality 20 marks, organization of the content 20 marks, clarity of the work 20 marks, knowledge of material 10 marks, neatness and overall presentation 10 marks, questions and answers 15 marks. So with all that, finally the results have come. Before I announce, I want to thank for this opportunity and also thank my EC members for their support and encouragement. I would like to acknowledge my research scholars Ragni and Radhika, the web manager and Dinesh from Cisco, and my friends and family members for their support. With all this together, cumulatively made the online conference happen at such a grand level. So the prize winners, as we announced earlier in the circular, the awards will be presented by Biomedicine and Cosbeer. For the Biomedicine prize, winners can publish a paper to be submitted within 3 months, and publication fee of rupees 6,000 will be waived by Biomedicine. The first category is theme 1, that is bioinformatics and system biology. The first prize goes to Pita, PG Vaishnava College, Arambakam. Second prize goes to Ajit Kumar, Bharatiyar University, Coimbatore. We go to the second theme. Second theme is artificial intelligence and healthcare. First prize goes to Indranil Manna, Napoya Deemed University. Second prize goes to Mukilan, Wales Institute of Science, Chennai. Congratulations to the winners. Sub-theme 3, Herbal Medicine and Drug Discovery. First prize goes to B. Chitra, Bharati Women's College, Chennai. Second prize goes to S. Sarada, Soko Ikeda College, Chennai. Congratulations to the winners. The fourth theme Advanced Biochemistry and Microbiology. The first prize goes to Thangappa VK, Nijalingappa Medical College, Palukad. Second prize goes to Renuka Sundari, Tigmat Pondicherry. For Cosbeer Award, the winners of the poster session will receive a check of rupees 2,000 from Cosbeer to address provided by the winner. First prize goes to Samira Begum, Napoya Deemed University. Second prize goes to Dhanashri. Yeah. This is Kasturba Medical College. Congratulations to the winners. Hospital trustees have also announced special cash awards of rupees 2,000 each for the following persons. Radhika and Ragini for coordinating with the organizing secretary for preliminary and preparatory work throughout the conference. We have also announced 2,000 rupees cash award for the web manager IABMS and Dr. Dinesh, team leader in Mentor Infocom Private Limited, IABMS Cisco WebEx Virtual Conference Manager. This is for their commitment and dedication in planning and execution of the delivery of the contents for the past 3 days. We thank wholeheartedly for the trustees for their sponsorship for the entire conference. Okay. They've given us the platform, hospitality, and e-poster prizes. Thank you for the, once again, thank the trustees. So now we go to the another interesting part, that is the feedback of the conference. So now we just now received feedback for the conference. There is some information about the conference first and feedback. Conference registration through the website was 274. Conference registration through the Google Forms were 126. Total abstracts received were 76. E-posters approved and received were 15. E-presentation approved and received were 56. And disqualified was 5. Skip minimum, okay? And you see the views, how many people. This slide shows you the overall conference page content. It's very interesting for the viewers to have a look at how the views went through for the entire session. It is available in the net. Now we got feedback from our participants. Now it's a hot news coming. So they have graded us as 4.6 for 5. So we all should really feel happy and very thankful for our generous audience who have given us a very good rating. Thank you. And here comes the fresh news from our COSBEER. They have given us very good feedback. They are happy with. All the COSBEER members were watching this conference for all the 3 days, and the feedback has come right now. This is for your for you to view and appreciate. Thank you very much for this opportunity.
A very good evening to all the participants of the conference. This is Dr. Srikumar Chakravarti from Masa University in Malaysia. As much as I have this opportunity to talk to you right now, I am also glad that I had the opportunity to give the keynote address a while ago. And it is heartening to see that there are so many participants who have participated in this event, and with so many papers and this online conference reaching so many countries, it is a great honor that the 41st annual conference of the Indian Association of Biomedical Scientists is a grand success. Even though it is a virtual conference, this conference for the past 3 days on 12th, 13th, and 14th March has already garnered such a wonderful response as we saw in the previous presentation by ma'am, and it shows a wonderful rating of 4.6 out of 5. On behalf of our university, Masa University, which is a co-sponsor and co-participant, I would rate it 5 on 5 or even more if I had the chance to do so. For the simple reason that this conference has reached out to so many people in every nook and corner, despite it being online and during the time of the pandemic where we couldn't step out from our homes. And we all could actually access the content from the safe confines of our homes and yet get updated on all the latest scientific developments in medicine or bioscience or pharmacy, health sciences, nursing, and so many other fields. And we were able to mobilize the research knowledge from all the presenters and also all your various innovative solutions. So we as part of Masa University are really glad to be part of the organizing committee with the IABMS, and we would like to thank the organizers, Dr. Padmini, and the president, Dr. G.K. Pal, the treasurer, Dr. Hema Malini, and our organizing liaison officer from Masa University, Associate Professor Dr. Vetriselvam Subramaniam. I'm sure they've been working day and night for the past few weeks. As ma'am had said earlier, Dr. Padmini has been working for a few weeks almost every day. And the same thing with all the organizing committee members. So I understand that a great effort has been put in for a conference of 2 days, and this has helped us all to actually get to understand the recent trends in biomedical research. And the goal of this conference was to build awareness around all the advancements and the sub themes which you have mentioned in the brochure. And the sub themes have been apt, including some latest themes such as artificial intelligence. And some of the papers have been of such good quality that we have even given awards to them. So, thank you once again for allowing me to be a participant and also an opportunity for me to give my feedback on the excellent and successful running of the conference. And we hope that this continues in the years to come. We have created an impact by starting the conference online for the first time, so that in future it could be both online and a physical version, or we could have multiple versions of it as well. So thank you so much, and we hope that all of you have been benefited from this as much as I have during these 2 days. And once again, I would like to thank the organizing committee for all your unstinted hard efforts. It has not gone in vain. It has been a great and grand success. Thank you very much once again.
Good afternoon, everybody. Firstly, congratulate all the members of the conference, especially Dr. Padmini and others, for making this conference very grand success. I know that organizing the virtual meeting is a very difficult task. Especially, I am totally impressed with the 2 sessions running in one platform is very appreciable, and also I like the way it was organized. And I'm glad to see that rating also excellent by the participants. I also thoroughly enjoyed the presentations. I thank once again that Padmini and our organizing committee members for giving me the opportunity to participate in the meeting. Thank you one and all.
Good evening to all of you. This international conference was organized by IABMS with a co-partner of Masa University, Malaysia. This is the first time IABMS collaborated with an international university with a new platform. The conference was officially welcomed by Organizing Secretary Professor Batmini. After the welcoming speech, the conference was addressed by the IABMS President G.K. Paul. National and international speakers actively participated in this conference. The national speakers, Dr. Sanmukha Sundaram, Wales Institute of Science and Technology, Chennai; Dr. Hema Malini, Kuwait E. Millat Government College for Women's; Dr. Subrata Ghosh, Bardwan University; Dr. Surajit Patak, Chettinad Hospital and Research Institute; Dr. Vijay Kumar Kutala, Nizam Institute of Medical Science; Dr. Tirunala Sundari, ex-president of IABMS; Dr. Ilavarasan, Ministry of Ayush; Dr. Uma Mageshwari, Simtas, Chennai; Dr. Sivasankar, Nanda Siddha Medical College; Dr. Sudhakar Manonmaniyam, Suntaranar University; Dr. Suja Bharadiyar University; Dr. Manjula Shantaram, Mangalore University; Dr. Venkataraman Tipsar, Chennai; Professor Rajagopal, Chennai; Dr. Vinod Bhagavat, SBH Government Medical College; Dr. Batma Kumar, University of Kerala; Dr. Ahmad Basha, Chettinad Hospital and Research Institute; and Dr. JLN, CEO, Ministry of Ayush, India. The international speakers, Professor Sri Kumar Chakravarti, Director of Research, Masa University, Malaysia; Dr. Ikapol Venkat, Mahasarakam University, Thailand; Dr. Saumitra Pal, Boston Children's Hospital and Harvard Medical School, USA; Dr. Shivkanya Floria, Ames University, Malaysia; Dr. Anandaram Devaraj, King Khalid University, Saudi Arabia; Dr. Lee Po Foong, Uttar University, Malaysia; Dr. Sriram Narayanan, Pfizer, USA; Dr. Sayatan Maji, University of Pennsylvania, USA; Dr. Nagalingam Varnakulendran, Eastern University, Sri Lanka; and Dr. Srinivasan, University, Uttar, Malaysia. All speakers delivered their talks very informative and focused on current and future biomedical research. I congratulate all prize winners and the participants. Also, I thank to organizing committee members, IT teams, sponsors, and other supporting staffs. Without your support, we never succeeded in this event. With a great immense, Masa very much appreciated in this international collaboration, and the future expected the same kind of collaborations. Additionally, IABMS going to have one more international conference, collaboration with Einstein Academy, Malaysia. in May 2021. With this, thanks to everyone for attending this conference. I am very sure we'll meet again with the different scientific occasions. Once again, thank you all.
Thank you, Dr. Vettichelvan. We are coming to the end of the conference. I just have 3 announcements to make. First announcement is that our editor-in-chief has agreed to bring out a special supplementary issue of the IAB, this biomedicine, with the abstracts and the papers. That is the first announcement. Second announcement is we have given feedback form for all the students, and they have to fill in the feedback form, and then they will get their e-certificate. That is in the process. It's already ready. Once you give the feedback form, the e-certificates will reach you. The third one is the prize winners, cash award winners. You have to send your address, to the EABMS mail, so that we'll get your address and the cash award will be sent to you as check, which you can encash it in your place. These are the 3 main announcements, and we are in time. We have completed the program in time. We started in time and we have completed in time. Thank you for all the participants, and thank you for everyone once again. And we will now stand up for the national anthem.